to everybody, and you have exceptionally good central facilities. All these things, and you have a team, instrumentation team, you have an engineering team, you have all these teams. All these things are his vision to have something that institution has everything that it needs to function absolutely very efficiently. Money was certainly better, and he has even started a fine biochemical central facility to everything. This vision is not something that can be matched. Seen many institutes, been in institutes, this is something that cannot be and has not been replicated since. So here are some photographs. This is going way back to 1983, where Professor Pushpa Bhargava with Dr. Sindhu. The last slide is the IICT director then, towards the end, and that is here. That one. And here is our then president, Gyani Jail Singh ji. And this is our 1986 when the 1987 when the institute was inaugurated. You can see then Prime Minister Rajiv Gandhi. I don't think we need to introduce people like them, and NTR and Nandamuri Tarak Ramarao. All these people were on the stage, and along with, of course, Professor Pushpa Bhargava. You've seen him in the previous picture. And here is the first Republic Day that has happened from that place. This was also in 1987, and this is Professor Pushpa Bhargava at that time. And here is the CCMB building after it was made in 1987. And here is the sculpture which we hardly see nowadays because of the trees in front of it. But I was told by somebody, I think Lakshmi, that it is essentially Sleeping Beauty or Sleeping Lady, and made by M.F. Hussain. So this is another thing about PMB, is that he had mixed both, like, he's of course scientific visionary, which is known, and his own scientific achievements are amazing. I think he had a back-to-back Nature papers in 1 year. 1979, if I'm not wrong. He had 2 Nature papers in a row. So, but he was very interested in arts, and he nurtured it, and you can see that in every nook and corner of CCMB as you walk through. There are paintings at every corner of the CCMB, and you know, we had, I believe, performances from all major musicians as well as dancers of the time, and then we had visits from Nobel laureates to political leadership to everything. So, you know, CCMB, when I first visited CCMB in 1991, the first glimpse of this place is like you're completely awestruck by it. That is the feeling that you get when you enter the place. You don't feel it is, I mean, I was in IIT prior to that. I was in IIT Bombay, but this was different, completely in a different world and a different level. So, lot of credit and a lot of things to, a lot of respect for the man who has built this. And it is, it's my pleasure and fortune that I'm actually sitting and talking about him as one of the directors of this great place. And I would like all of you guys to, today I would like these scientific activities to continue, and it will end with the culture program at the end. I really don't want to talk more other than to show another visionary who's also passed away in 2017. So this is Dr. Lalji Singh, who's our 4th director. He passed away on 10th December in 2017. Is another person with amazing vision. We are today celebrating Bhargava, but PMB, but we also have to remember the contributions by Professor Lalji Singh, who's founded in one sense CDFD, LACONS, and CRF. All these things came under and during his time. And he's also the one who's kind of standardized the DNA fingerprinting. The whole credit goes to him as far as that coming along in India is. So the last photograph I'm going to show is something that was taken in 2015 or 2016, I don't recall, but when all these directors came to visit CC, with the exception of Professor Sharat Chandra, all the other directors of the time are there in this photograph. You can see Professor Mohan Rao, PMB, Balu, and Rakesh, of course, and Lalji Singh. So in this kind of a photograph cannot be replicated because two of them are no more with us. So with this, I would like to thank you guys. I would hope that all this, there will be some amazing scientific activity and interactions today. Thank you.
अरे यार ये थोड़ा बैलेंस करो. Thank you, Director. We shall now move on with our first alumni talk. Our first alumni talk has been given by Dr. Rana Anjum. Dr. Rana Anjum completed her PhD from Dr. Ashok Karslat, and then she moved on for her postdoctoral training at Harvard Medical School, where she worked upon MAP kinase and PI3 kinase. Further, she worked at Ariad Pharmaceuticals and AstraZeneca Pharmaceuticals. And currently she is the director of the oncology clinical development at GlaxoSmithKline, GSK. So I'll be handing over to Dr. Rana now for her talk. I hope she's online and she already has joined us.
Hello, can you hear me?
One minute, madam. उधर से कराओ ना.
Can you see my slides and can you see me?
Ma'am, I can see your slide.
हेडफोन सी है करेक्ट है.
अभी पहले भी कुछ
ऑडियो कनेक्टेड है सर, क्योंकि स्पीकर में साउंड आ रहा है. एक बार मैम को कुछ बोलने बोलिए. वीडियो में क्या आ रहा है काम का आवाज आ रहा है.
You can speak.
Good evening, Dr. Rana. Can you hear me?
Yes, I can.
Okay,
Can you hear me?
I'll be handing over the session to you now.
Okay. Can you hear me okay?
Yeah, we can hear you.
And you can see me?
Yeah, we can see you.
And my slides as well?
Yes.
Okay. Should I start?
Yeah.
Good morning, everyone. Good morning, Director CCMB, staff, and students. I am really excited to be here in front of you. I'm very humble and very honored to be presenting on CCMB Founders Day. I wish I was there in person, speaking right to you in front of you, but I think pandemic has taught us new ways of working, and it works now that I speak from so far, but I'm always close to you there. Hopefully, I'll come and see you sometime soon when all this pandemic is behind us. I never presented a talk like this, summarizing my entire career. And this is an attempt I made. I just made few slides just today. And so apologies if it doesn't cater to your needs, but I thought it would be nice to actually talk to the grad students about my career journey. and give some understandings and challenges and perspectives of where I started and where I reached, and like. So I thought I'll give a brief introduction to each place that I had been, as well as some background on the areas that I worked, a few challenges that I have overcome, etc. So before I jump into my next slides, I thought I would start from here. As Director CCMB had said a lot of good things about CCMB. This is the place where I started my career as well. And I'm always proud of CCMB, proud of everything that CCMB has done for me. So before I start going ahead from where I landed from here, I thought I would give, I'm very excited to see Balu. I'm so excited to see you. Hi, Balu. So it was he who recruited me. So, so before joining CCMB, I thought I would let you know what were the challenges that I was facing and where was I. So, like most of you, my dream was to pursue my career in medicine, and as most of you have written medical entrance exams, had written MSET. And I narrowly lost the government seat. And my parents didn't want me to pursue my career in any private college, even though the fee was so minimal when I think about it now, because it was against their principles. And so I had to quickly change my career trajectory. And I started my bachelor's in biology and then continued my master's in biochemistry from Osmania University. And I really loved it, and I secured a gold medal after my 2 years. And that was when where I need to go. And then I heard through my professors at Osmania University, the different institutes that are in India, where I can be part of a PhD program. So I applied to TIFR, IISc, CCMB, etc. And my father at that time had said that I cannot go out of Hyderabad to continue my career. So all I have to do was to be in Hyderabad and continue my studies. So I wasn't allowed to go to IISc Bangalore. So as you can see, my family was very well educated, but they were traditional and conservative as well. And they didn't want me, a proper Hyderabadi girl, to study outside of Hyderabad. And so I came for interview at CCMB. And I remember vividly my entire interview process, all my interviewers, even the questions I had answered. And then I went back home and I told my family that at that time, it was not that they would know where I have been applying and, what is the progress in terms of PhD applications, etc. So I reached home and I said, well, I attended an interview at CCMB. It went great, and I'm sure I'm going to get a seat. And the next day, we were I think we were called, and I remember in the director's office with Balu and others, meeting other 6 CCMB students for PhD program. So that's where I started. And I feel I was really fortunate to have been selected at CCMB. As I have mentioned here, it was like a launching pad for my career. And at that time, I was interested in the biology of cancers, and I joined Dr. Ashoka's lab as a PhD student. And at that time, apoptosis or cell field, and I wanted to learn and do more in that area. But something that intrigued me was the culture of CCMB, the science that was done at CCMB. something unique was that CCMB had such diverse areas of science. You name it, I can count them, but I don't want to miss anything. But it was everything in a very small place. A great culture, exposure to lot of good science, very talented staff, etc. And so now from here, I have to build my career, and I understood that I have to go out, I have to do my postdoc, I know that it's really challenging for me, as you heard that I couldn't go out even to do my PhD at IISc. So getting out of Hyderabad and going abroad, especially to US, was like a dream. So my way of overcoming this challenge was to carve my way around it. So what I started doing was to start attending as many meetings and conferences as possible. I remember my first meeting. I had gone to Tata Memorial Hospital in Mumbai to attend an apoptosis meeting. And my father has sent my younger brother along with me, who was a high school student at then, because he didn't want me to go alone. And then there was another conference after that, and my brother tagged along with me. But I knew that I had to do something. I had to prove that I can go out, I can be independent, and I can do all that myself without any help. And I applied to for this Young Investigator Award. This is something from American Association of Cancer Research, one of the prestigious award. And I think at that in '98, I believe there were 4 or 5 students throughout the world who were selected, and I was one of them. And I was given $2,000 as a grand prize, and I got tickets to go and attend. And was completely shocked at the culture and everything, but was very excited to present my work and to meet a number of people. So what I did was, so at this time, going all the way to New Orleans, my brother couldn't accompany me, and it was all by myself. I was all by myself, and was just trying to network and trying to see as much as possible in that short time. So I went and visited Baylor College of Medicine, MD Anderson. Then I went to New York and Boston. Luckily, I had some friends at each and every place. And so that was nice meeting them, and also understanding different institutes and trying to make sense of how I'm going to be here someday. So it's all visioning and planning. And then I came back. I went for another meeting at Cold Spring Harbor in '99. Again, just showing that I could confidently go out and come back by myself. Went for some other meetings, won several Best Poster Awards, etc. So this is how I try to carve my path to where I had to be someday, by networking, by meeting people, by attending conferences, etc. And then finally, when the time had come for me to leave Hyderabad, it was very, very challenging. It took 1 year for me to convince my family that I had to leave. And that was completely against what they wanted and what the big family wanted, because they never wanted me to go all by myself. So my flight tickets were postponed or canceled several times. And unlike at that time, there was 9/11 as well. So finally, after trying for the entire year, I could convince my family that I am leaving and I'm coming back after 2 years to after I finish my postdoc. And I'm really thankful to them for trusting me and sending me. So that's how I left. And one of the people I had met in at ICGV meeting was Professor John Blennis, and I had applied for a postdoc in his lab and was selected. And so I landed in Boston at Harvard Medical School, and my aim was to complement the cell death experience that I had here, the apoptosis and cell death signaling with cell survival signaling at the signal transduction group at Harvard. So this was a wonderful time. It was a time when the signal transduction pathways, especially the MAP kinase and PI3 kinase pathways, were being unraveled. So I was one of the finest lab with such pioneers in the field. So it was the group, signal transduction group of Professor John Blennis, Professor Luke Cantley, and Ben Neal at Harvard that I was part of. And it was great to be there in those bi-weekly meetings where all these new discoveries were being made. As you must have heard, the signal transduction pathway downstream of PI3 kinase pathway, the TSC complex, Richter and Raptor and mTOR, those all discoveries were made when I was there. So I was in this lab of 20 postdocs, a huge lab, which had its own challenges. But the biggest challenge for me was the weather. Of course, I had never imagined what cold can be. And I had reached there with just a leather jacket, thinking that, it's just cold and I can, be fine. But I hibernated for about 6 months. And then it was, I think, summer that I finally thawed. But I was really glad to be in this lab. And the main work in Professor John Blennis's lab was the area of self-survival signaling, especially the RISC family of protein kinases. So, so as you see from this figure in the middle here, the RAS-MAP kinase pathway that is activated in response to the growth factors such as EGF, etc., serum for esters. It leads to activation of RAS-MAP kinase pathway, RAS, RAF, MEK, and ERK, and finally leading to risks. As you know, this is one of the major pathways that is deregulated in cancers. So we were looking at mechanisms of signaling downstream of all these proteins, the mutated RAFs, mutated MEK, ERK, and then finally risks. So this RISC family of kinases are 4 proteins, very highly homologous, with very complex way of activation. With it has the C-terminal domain and an N-terminal domain, etc. So our lab was looking at the substrates of RISC family protein kinases and how this signal pathway that is culminating in RISC is actually collaborating or impinging on the parallel pathway, the PI3 kinase pathway. So as you can see from here, there's cooperative signaling between RAS-MAP kinase and PI3 kinase pathway. And as you know, these 2 cell signaling pathways are the major controllers of cell survival and cell death signaling, and are the ones that are deregulated in most of the cancers. So not going too much detail into the work that I did there, I thought I would just give some highlights. One is that, as I said, that we were looking at the cooperative interaction between MAP kinase and the AKT pathway in regulating cell proliferation and cell death. And so this is the one that we showed that both MAP kinase and PI3 kinase dependent phosphorylation of this protein complex TSC1 and 2 controls mTOR signaling. And then we finally found a specific protein that is activated by RISC in TSC2. And the other part that I worked on is the tumor suppressor death-associated protein kinase as a target of risk-mediated survival signaling. So what we showed is DAP kinase has a specific phosphorylation site. It's a tumor suppressor, but it's the risk-mediated signaling that actually inactivates DAP kinase and leads to attenuation of cell death and thus enhances survival signaling. So using all those biochemicals and in vitro cellular assays, we were able to uncover a number of targets of risk, as well as this mechanism of cooperativity, as well as linked to oncogenesis. So I was there for a couple of years, and then always the cell signaling lab, you have a protein and you need to find the phosphorylation site, and then you are actually dependent on the proteomics group. So we had a really nice proteomics group at Harvard, just like the next floor that I used to work in. It's a lab of Steve Gigi, who was a professor in the same department. So he had this mass spectrometry core, and that was like a sister lab for us, because all the work we do in finding out phosphorylation used to be actually helped by the mass spectrometry facility. And so what I did was to talk to people in the lab and try to build collaboration. So that was a huge lab as well, but it was just really focused in mass spectrometry and proteomics, with people in the group lacking cell biology experience. So I thought that would be a great opportunity for me to collaborate with them. And so I could learn proteomics, and I could teach them cell biology experience with working with a number of cancer cell lines and models, etc. So this developed into a nice collaboration. I stayed in the lab for 1 year, but it was highly productive. We developed highly sensitive multiplex kinase kinase assays that you could take nanogram of protein, and you could detect phosphorylation of, say, 90 different proteins in a cell extract using this phosphoproteomic approach and using LC-MS quantification. And this was also, this on the left, I'm showing the schematic, and on the right are different agents or cell lysates, HeLAs or HEC293s with different constructs overexpressing different oncogenic kinases. Using their specific inhibitors, we were able to recapitulate that in cell lines and cell line models. Not only that, being part of Gigi Lab, I got to interact with many other labs outside of Harvard, and actually collaborate with them and be part of their discovery. So that was a nice time. And again, mentioning this because you have to actively seek opportunities, and it's this unique opportunities that help you develop. So that was part of my postdoctoral experience at Harvard. So summing here, my 5 years or 6 years that I spent at Harvard, I'm listing some of the challenges. Of course, getting grants was a challenge. And when I reached there in 6 months, I was told that that I would be supported only for 6 months, and after that, have to get my own grant if I have to pursue my research further. So that was a nice pressure, but you have to get a grant to continue studies in the lab. And then there is a high tough competition, 20 postdocs, each trying to work around the same areas in the same field, and so you need to find your own particular niche to develop further. And then, as I mentioned, key learnings is like seek opportunities as much as possible, be agile and adaptable, and learn to lead and influence. Networking and collaboration are really key. And again, when you are a postdoc, that's one challenge and you always struggle with, which is finding the right career path, because you believe that you have to find a place in academia, and you spend a lot of time trying to build your resume or trying to look for a position. And then there is industry as well. And there are, I think, opposing views as to on the pros and cons of each. But I think each is unique in its own way. And so I did spend a lot of time as well trying to decide where I would go and what is my next step. And that actually led me to lose some time as well, trying to make up my mind. But again, trying to network with companies as well as different academic institutions, other labs outside Harvard. The great thing about Harvard is it has lots of institutions affiliated to it, and you can meet a lot of people, clinicians, postdocs, even faculty, to discuss and talk about your career and get their feedback. So I finally decided to join industry here with mixed feeling, whether it would be the same, whether it would have the same challenge as academia, especially the science. Is it going to be the same? Because I have heard different things about industry. But when I joined, I was very excited, and I actually found no difference, or almost little difference, between being either in academia or industry. It was almost the same, but with a twist. Here, it is all about the patient. It's all about getting the drugs into the clinic. So my first position as a senior scientist was at our Ariad Pharmaceuticals. So this was a small company in Cambridge, Massachusetts. When I joined, it was a company of around 50 people. And when I left, it grew to 500 people. So this was a company that was actually founded in 91, and their focus was targeted therapies and trying to unravel mechanisms of resistance to cancer therapeutics and trying to develop best-in-class inhibitors. So pretty good scientific focus here. And me being so much into cell death and cell survival signaling, I think this was something that really excited me. The focus and the thought of taking a drug right from the chemistry all the way into the clinic. So as you all know, drug discovery is not really easy. It's 90% of the drugs that actually develop fail, and there's a lot of attrition, and only a small percentage reach the clinic. When you actually consider all the time from developing your leads into developing into a drug, there is a lot of attrition. So here I just put together some schematics to show you that development starts with discovery. And again, this is all basic science, trying to find a niche, your drivers of your cancer, your targets, and your target validation. And then that leads into a lot of preclinical research, trying to identify a target and trying to see if it's the right one, it has the right properties, and then developing a drug that goes through a number of stages, as I have mentioned a few here. You have to go through target validation, a thorough target validation of whatever target you choose. And then you develop your compounds, and you screen in a different ways, in high throughput screening, in selectivity screening, and using a lot of computational approaches or structural biology approaches. And then going into a number of secondary assays, testing the in vitro and in vivo activity of the molecule on the target. And then followed by in vivo analysis, a number of models disease models as well as engineered models, and then going into early safety studies in mice and monkeys and toxicity studies, followed by preclinical safety and toxicity package. So it's a lot of work, number of years of work, and it takes about 12 years to get a drug from actually trying to find a target to finding leads and hits, and then going all the way into development of a new drug. So when I joined Ariad, they had an mTOR inhibitor, that is radiofrolimus, as well as BCR-ABL inhibitor, Ponatinib, that was in clinical development. And this company was trying to find other novel targets and again, targeting cancer through some of these novel approaches. So Ponatinib is a BCR-ABL inhibitor. And as you know now, or even then, it was in clinical development, was in phase 2 for CML as well as Philadelphia positive ALL. So I joined at this time, and I was part of a number of discovery efforts. Not only these, but some new targets, as well as some of the translational studies of Ponatinib. So when you are in a small company, you have to wear a number of hats, and you can do almost anything you like if you have the dream or the wish to do a number of things. So I was interested in translation, for example. So I could actually be part of the translational activities, even being in discovery. So I got to learn a lot in the small place at Ariad Pharmaceuticals. I'll go through some of the targets that we discovered. One particular molecule or drug now, that is Brigatinib, that was developed all the way from finding a single molecule from a really low throughput screening chemistry to all the way to FDA approval. So at least I can brag about having one drug approved based upon my efforts. So, and that is very satisfying that you have something that you have brought to the clinic or to help the patients. So non-small cell lung cancer, as you know, has many different drivers. For example, EGFR is seen in about 15 to 20% of adenocarcinomas, followed by KRAS, which is another big chunk. And then there are a number of small aberrations like ALK, ROS, and RET, HER2, etc., and mutated RAF that leads to the development of adenocarcinoma, especially non-small cell lung cancer. So at that time, a group in Japan had discovered the fusion protein EML4 ALK as something that leads to the development of non-small cell lung cancer. So this fusion protein leads to deregulated signaling, and especially it impinges on the two survival pathways, leading to constitutive activation of RAS-MAP kinase pathway and the PI3 kinase pathway, again leading to cell survival and cell growth. So this is the pathway that is activated in these cancers. And this EML4 ALK-driven non-small cell lung cancer is seen in younger population, in non-smokers actually. And that was a new target that was discovered. And I think this particular target led to a drug in really short time. So this was a really fast approval. And at that time, Pfizer was having an inhibitor, Crizotinib, which was actually a MET inhibitor. And they repurposed it for ALK-driven cancers. And Ariad, being a small company, was looking for ways to get into the field, and Ariad was really good at finding best-in-class inhibitors. So when Crizotinib was used for ALK-positive lung cancers, patients came out with resistance, especially to gatekeeper residue. And at that time, that was the challenge and opportunity that Ariad picked up. And we developed a best-in-class inhibitor for ALK-positive EML ALK positive lung cancer. So that was a long journey, but then we have a number of papers, and we show that the ALK inhibitor Brigatinib is very potent and can overcome resistance to first and second generation ALK inhibitors. So after crizotinib, there were a number of other next gen inhibitors like ceritinib and alectinib, and Brigatinib was really potent. And based on its potency, this was approved for ALK positive lung cancer. Another project that I thought I should touch is for Ponatinib. So Ponatinib was being developed for BCR-ABL cancers, CML and ALL. And at that time, it wasn't approved yet, but we were trying to find other indications where Ponatinib would work. That's because most of these drugs are usually multi-kinase inhibitors. So Ponatinib did have activity to a number of other kinases, including KIT and PDGFRA. So we thought we would repurpose it for GIST, or gastrointestinal stromal tumors. So this particular tumor is one of the most common subtype of human sarcoma. And in this tumor, there are about 90% of cases where there is a gain of function mutation of KIT or PDGFR alpha, and there is an unmet need for patients in this particular indication. The reason being that none of the drugs that are approved work fully here. For example, this is the KIT structure, the different domains of KIT. So there are, first of all, there are primary mutations that come up in GIST, which is mutations in exon 9 and exon 11. And so the first line agent, let's say imatinib, the drug that is used in the first line is imatinib, and which is very potent to inhibiting exon 9 mutations, insertions, and point mutations, exon 11 insertions, point mutations, etc. But if once the patient is on imatinib, at least 80% of the patients develop drug resistance in about 2 years. So, and they come up with the secondary mutations that are clustered in ATP binding pocket and activation loop. So there is V654A mutation, this is the gatekeeper mutation, and the number of A loop mutation, they all come up. So a particular tumor can have as many as 5 or 6 different mutations. So when imatinib doesn't work here anymore, as you can see, it is all red or resistant, then the second line drug that the patient take is sunitinib or Sutent, that actually is very potent against the ATP binding pocket mutation, but is ineffective against the A loop. And so patients again come up with resistance. And then the third line agent is regorafenib from Bayer. That is again inhibits few but not all. And the problem with both sunitinib and regorafenib is that they have this VEGFR activity as off-target activity. So because of this VEGFR2 activity, you cannot optimally dose the patients, and therefore, even though this is mentioned or written as green, that there is actually resistance because you cannot optimally dose the patients, and therefore you cannot actually inhibit this mutation. So patients that are suffering from GIST are in need of very effective treatment after they finish their treatment with imatinib, sunitinib, or regorafenib, because they have no other treatment after that. So that's an unmet medical need, and Ariad was trying to repurpose Ponatinib into this indication. And we developed this data package in such a small time to initiate a phase 2 study. This paper was published again in Clinical Cancer Research. But okay, here I show some of the data that was in the paper. Really nice potency data for Ponatinib. You can see in green bars here with comparing with other drugs, Imatinib, Sunitinib, Brigatinib. Sorry, here is Ponatinib showing low nanomolar activity against most of these mutations, ATP pocket and A-loop. So just showing a little bit of data in mouse models where Ponatinib at 30 mg is very effective, and this was taken in clinical trials. And this is the data from 3 patients showing a disappearance of tumor lesions where there was resistance when they were treated with earlier drugs. So really good data package, but bad things happen sometimes, and this is something that is unreliable. Sometimes when you work with kinase inhibitors and in pharma, that anything can happen. So there was a crisis, and one of the trials that Ariad was leading in the CML space with Ponatinib showed some blood clots in patients that were treated with Ponatinib. And that led to the canceling of the clinical trial. With that, a lot happened. The company crumbled, and a big part of drug discovery was cut off to actually compensate what was left, because now they had to focus on the clinical trials that were ongoing. So the discovery was cut off. So really bad time for everybody. So this is something very common that happens. But for me, it was an experience that I had never faced before. It was a very challenging time, being laid off from a company, having worked so hard on a drug. It was when I left that the drug Brigatinib was approved, the drug that I had worked for. But again, with crisis, with challenges, there are opportunities. So always look for opportunities again. Look for those doors to open. With this, I joined AstraZeneca, and at AstraZeneca, so I'm just pointing at different projects that I led in drug discovery, in translational strategy, in clinical science, and in clinical development. So I went from drug discovery all the way to clinical development. Here as a senior scientist, again, just like in Ariad, leading drug discovery projects, new target ideas, new drugs to new targets, project teams, involved in a number of governance interactions, developed chemistry and small drugs for a number of solid and liquid tumors. I'll tell you how I started with some of these targets. And then with my interest in translational strategy, became a translational strategist on secondment for one of my projects, IRAK4, then looking at clinical biomarkers, predictive biomarkers, etc. So I was actually more interested in coming from drug discovery all the way towards clinical development, was passionate about now being close to the patients because I have been working on targets and inhibitors for a long time, wanted to see how it is applied. So that was something that I always felt, okay, what is beyond drug discovery? I want to see translational strategy. I want to see clinical development. And how do I build my path towards that? And it's not easy because everywhere they need experience. When you go into a clinical development program, they need somebody with clinical development experience. And you don't get clinical development experience unless you work on it. So it's a difficult situation. So I started working in preclinical science in clinical programs. So Savolitinib was a MET inhibitor that was in clinical development. So I became the bioscience lead for that program. So it's a clinical program, but then it's like science from the bedside to bench and back. So whatever was seen in the clinic, it was coming back to me to have a new strategy to test it again, in a number of models, and then again take the learnings from there and then apply it to the clinic or to the bedside again by altering the clinical trial or by starting something new by having a combination study. So we were studying combination of say MET inhibitor with an EGFR inhibitor, etc. And then managing preclinical budget, and then working with business development to out-license assets. And then from here, I moved into clinical development. I'll talk to it in a moment, how I did that. But one thing what I did was as when I reached AstraZeneca, I jump-started the KIT program, having all the experience from ARIAD. And at that time, development of a KIT inhibitor was still an unmet need. And so I took on this opportunity. And then in a short span of 3 or 4 years, from all the way from chemistry to the development of a potent and selective KIT inhibitor, something as you can see, AZD3229, again, very potent against all these different competing molecules, imatinib, sunitinib, regorafenib, and there were 2 more molecules that were developed when this program was going on. So published a number of papers. This became my pet project because in the end it was left to me, and the priorities had changed, and then AstraZeneca decided to out-license this asset to another small company because of changing priorities. And I was just scrambling to get this published into a nice journal before handing it over to whoever bought the drug. So a very challenging time trying to assemble a group of people to actually put together the manuscript, but it all worked really well, and we got a nice paper, 1 or 2 nice papers on this drug. And then this was another project that's worth mentioning, that is IRAK4 for ABC-DLBCL. Again, ABC-DLBCL has a worse outcome with R-CHOP chemotherapy, and there are some activating mutations in MYD88 that occur in say 30 or 40% of ABC-DLBCL patients. L265P is the most prevalent one. So as you can see, we were trying to develop an inhibitor for IRAK4, and that can be used in combination with BTK inhibitor, acalabrutinib, that AstraZeneca had. And we had a number of papers in this, but it was a very challenging target. Trying to dial potency and selectivity was very difficult. And so this project stopped. But I was in the savolitinib program then, and then I wanted to pursue further into clinical development, and it was really hard to move from preclinical science to clinical development. So this was one of the challenging points, I should say, and how I approached it. I applied for a position in clinical development, and I was given the opportunity, I was given the position, but I could only join after I finished a 6-month secondment. But at that time, the preclinical science would not have any open positions. So for me to leave the preclinical program and delve into clinical science, clinical program. So either I stay or I leave. And at that time, I had made a decision to leave preclinical science and join clinical development. So I took I had to leave my position and take up a semi-permanent position, for example, to join clinical development. So it was like a contract position. So my 6 years of work at AZ were just finished, and I took up a temporary position to join clinical development to finish that 6 month secondment, but then later joined as a director in clinical development. And that was a very challenging time. It was in the middle of pandemic. And I remember starting at 5 a.m. just to overlap with the UK timings because the clinical programs were based out of UK. But I made it work. So in the end, it's hard work, and what you want to achieve, you could achieve. This is an example to show that. And so after joining clinical development, I studied, I was in the capivasertib or AKT inhibitor program for prostate cancer. As you know, prostate cancer is one of the most common malignancies, and nearly all patients experience disease progression and develop this castrate-resistant mCRPC, and there is unmet need for novel treatment options. And so we were looking at capivasertib in combination with some of the other drugs, abiraterone acetate, in patients with castrate-resistant prostate cancer. So a number of abstracts there. But when I was actually in clinical development working on this project, I was approached by GSK, and I was intrigued with the immuno-oncology platform that they had. I started as an immunologist, and to get back to immuno-oncology was something I really liked. So I took up that opportunity. And there are a number of focus areas that GSK has, and immuno-oncology is one of its prime focus. And there are a number of, again, novel IO agents that are in testing at GSK. And here I am in one of the lung cancer studies, phase 2, phase 3 studies, testing a novel inhibitor along with a novel PD-L1 agent in lung cancer. So that's where I am currently. And the journey continues. I don't know what is next, but it's been really exciting few years in industry, and something I was very hesitant in joining, but I was very intrigued by the depth of science, and also something that gives you satisfaction of bringing drugs closer to the patient. So when I had made the decision to join, I was very hesitant when I left Harvard to join a company, because I was very academic oriented, and I thought I would miss that touch of science when I move into industry. But this has been very rewarding. It's not only great science, but also how it benefits people. It's nice to see that you're doing something that is benefiting cancer patients, and it's really a sense of gratitude. It gives you a sense of gratitude at the end of it, at the end of everything you do, and it's something that drives you. So I'm very much looking forward to continuing on this journey. And so this is my last slide, but I would like to acknowledge everybody, especially where I started, that is CCMB, all my ex-lab colleagues, my mentors, everybody, the graduate committee, the CCMB graduate committee that I was part of initially, Dr. Ashok Kar, and all the lab mates, Ashok Kar's lab, tissue culture lab, all South Wing ground floor people, the coffee mates, I should say, South Wing second floor coffee mates, and everybody that made this journey so successful. So thank you all so much. And everybody at HMS area at AstraZeneca and GSK, patients and caregivers, and my family and friends.
Thank you, Dr. Rana. That was a very engaging talk. We shall now take questions from the audience. I request anybody who has questions to come forward and ask those at the podiums that we have installed here on either side of the stage. Online audience, you are free to ask questions if you have any.
Hello, am I audible?
Yes.
Yeah.
Yes.
That was really good talk, pretty engaging. So
Thank
I have
you.
a very basic question, not necessarily related to your work, but more to do with your journey. So from what I understood, you taking up a postdoc was your way of establishing yourself in the international market, if I can say it that way. For somebody who's pursuing a PhD today, would you say that doing a postdoc is absolutely necessary to get into industry or any of the allied fields, or is it just an add-on? Could just a PhD suffice? Yeah, that is my question.
So will a PhD suffice to enter into industry is the question, right?
Yes.
Yes. Definitely. I think it's just an additional step of taking a postdoc, but I think you can get into a company after doing PhD, and nowadays companies do have postdoc programs, so you can actually join a company and do the research and make that part of your postdoctoral research if you like, or just do your PhD and then diversify in other areas of interest, and does not necessarily have to be a postdoc.
Thank you.
Sure.
Hi, Rana.
Hi, Manjula.
Yeah. So it was very nice knowing your story in detail. Well, and seeing you coming up, from 1995 onwards to, this heights is very inspiring. I'm sure the students would all have benefited, right? So I also think you, of course, knowing you personally, right? I also know that you're a devoted mother and wife and even a daughter, right? So how did you manage all that? Because I think that's a major question, right? How particularly women can manage both a successful career and a family. I think that would benefit the students.
Yes. Thanks, Manjula. Great seeing you, and a really nice question. It's a question that doesn't have a straight answer. I could say that it has been a struggle at every level, and nothing has been easy, except that there is determination and there is focus, and then you want to achieve something. But it was not easy, and it is still not easy, as you know me personally, and you know how I fought my way. If being in a traditional family, to leave everything and come to where I have been now, that was a struggle. It's a struggle even now, trying to balance life, family, and career. It is not easy, I should say, but I think when you have determination, you find ways, and you find niches to help you move forward. And it is the purpose, it is the work that you are doing that is so engaging and that is so important, and it's at the core of your thought that you have to do it. And I could say that it is that I have been fortunate to have a family who trusted me, and let me follow my dreams. But it was not easy, and it is still not easy, I should say. But it's just the determination that keeps me going.
This is Dr. Sir. Thank you very much for sharing your struggling story. Just I want a small thing like in case somebody wants to establish his career in drug design, but he has no experience in clinical trial or is not very. He's not very he's not into the clinical trial kind of thing. So what kind of advice would you like to give? Like, don't go for it, or like what kind of advice you will like? Mostly starting with in silico kind of work. So, yeah, what kind of
I could not hear the question completely, but I heard a little bits because you were a little bit away from the mic. But the question I thought is about drug discovery. If you are interested in having a career in that drug discovery, how do you go about it? Right? Is that your question, right?
Just adding to that, that person, if he does not have any experience in clinical trial kind of things.
Yes. Yeah. It is not very easy. It's not easy to get into clinical trials because I had struggled myself at AstraZeneca, for example, having so much of experience in early drug discovery and in translation. To go into clinical development was a different ball game, I should say. And I had tried for 2 or 3 years because I was passionate about being close to the clinic and trying to move from early drug discovery to translation and then finally to clinical development. It was hard. As you said, I had to leave my job to join clinical development. I gave up my job, and especially in times of pandemic, it was a very uncertain time. And there was a little bit of hesitation if everything is going to be as I thought, if I'm going to be moving from a contract position to a permanent position in this after this 6 months, it was just something that it was hard to decide at the time. So it's not easy, but again, I think there are ways. I think there you can join a small company that has expertise in drug development, for example, take up a part-time position, and then somehow try to converge into the drug development field if you're really interested. Otherwise, it's not an easy thing to do. I cannot hear you.
Hi, this is Saujanya.
Hi.
Your journey has been truly inspiring. I would like to know if there is a single piece of advice that you would give to your younger self.
Yes. There are lots of advices I would give my younger self. I think one would be to network as a PhD student. I mean, I had done quite a bit of knowing people outside my field, outside the country, in specific areas of your development, writing to them, attend as many meetings as possible, try to network and meet people, try to understand which areas you would like to pursue. And one advice, advice I would give is to consider the time of PhD seriously, which later I thought, and I thought later after finishing my PhD that I should have actually worked towards shortening it a little bit, giving more time to do things after my PhD, but being very focused and trying to finish it as soon as possible, so that you can venture into to more interesting areas after your PhD. So So those are all those advisors that I think I have from my own experiences.
Thank you, Dr. Rana, for being with us today. As a token of our gratitude, we are sending you this small souvenir.
Oh, thank you so much. That's I'm really, I'm so overwhelmed. Thank you so much. Who did that? Such a nice one. Thank you. That's really nice of you. I appreciate it. Thank you so much.
Thank you, Dr. Rana.
Thank you. I see a question come through the chat.
Okay.
I think the question is what specific skills you need to have to develop your career in the industry when compared to academia. So I think, when you do your postdoc, you are actually working towards either becoming a faculty or pursuing a career in industry. So I think there is nothing different. I think the goals are the same, networking is same, and it's just the opportunities that you need to see when you are trying to develop in industry versus academia. So in sometimes you see all these big exhibitions come up and like industries presenting their work. So I would say network. Try to attend as many such meetings as possible. Sometimes they come once a year into specific cities where number of industries are putting together an exhibition. So of what different areas of development are there, or drug development the industry is putting together. So it is good to attend those kind of meetings. It's just like keeping an open mind. Networking is the key. And I think being open to either academia or industry would be great. Whichever opportunity you could come across, just embracing that would be great.
Hello. Thank you so
Hello.
much, Dr. Rana, for sharing your experience with us. And we shall now move on with our. Thank you so much.
Thank you so much. I wish you all the best, and I'm very excited that one of my juniors, Suresh, is presenting. I wish I was there as well, but I wish you all great luck in your PhDs, and I hope you succeed. And thanks again for inviting me.
I present thank you on behalf of Dr. Suresh.
Thank you. All right, thank you.
So I'll announce a short tea break for half an hour, and we'll rejoin our sessions after tea break. Yeah. Thank you.
Thank you.
I request all of you to be back by 11:10 p.m.
Yeah, Madam.
Very good to see you.
She's saying hi.
Hi. I can't hear her.
Let's switch
Okay.
this thing.
Okay.
He's left already. Okay.
See about crack. Apply lotion every day. Nothing happens.
Good morning. This is not working. Good morning, everyone. I welcome you all again. We shall now have the second of our alumni talks by Dr. Suresh Chintalapati. So Dr. Suresh has completed his PhD under the guidance of Dr. S. Shivaji, followed by postdoctoral training at Sanford Burnham Prebys Medical Discovery Institute. He was the chief strategist and member of the board at Genome Institute of Molecular Medicine and is currently the president and CBO of Triangulum Biopharma. I'll be handing over the session to you now, Dr. Suresh.
Thank you, thank you. So the year was 1991, and it was a winter morning. And at the school, the class was getting ready to study an old classic from 1876 by Mark Twain, The Adventures of Tom Sawyer. So the teacher actually had a very interesting way of keeping the class engaged. She used to make each student read a paragraph, and all the students used to start discussing, the language from the 19th century, grammar, written communication style, so on and so forth. And there's a very tall, skinny guy, thick glasses, well-oiled hair. He used to sit maybe 2/3 of the way, probably the third or fourth row from behind. And the moment this process started, he used to start counting the number of students ahead of him, and then count the number of paragraphs, identify the paragraph he actually needed to read. And he used to practice the heck out of himself to read the paragraph. And he used to hope that in his haste, he didn't miscount, because then he would have to read a different paragraph, and he was scared of that. This boy couldn't read. He had a lot of difficulty reading, and of course he didn't want to be embarrassed in the class not being able to read. So he went on, and he was following the norms of those days, and every student who goes through school would be expected to give the engineering and medical entrance examination. The boy went on to do that. No surprises there. He didn't qualify. Those days, I don't know how it is now, but those days you were tested for how much you could read and how much you could remember of what you read. Very few, if any, logic questions in the whole exam. So he went on to do his bachelor's in one of the local colleges, and his whole idea of joining, once he started his classes, his goal was
to actually read and comprehend. That's all. He didn't want to fail his exams. His fate actually turned to the better when two students in the class actually one day came to him and asked him to, explain some concepts that they were finding difficult to understand and comprehend. So he explained the concept, and the whole process went on for a few weeks, and the whole process was working out really well. From that point on, the job of these two students was just to take every single detail as notes in the class and come back and read back to this guy so he could explain everything to them. The boy went on to top the college in the first year, and his name was actually used in the prospectus of the college, advertising how well the college did and attracted more students to the college. Importantly, this boy could actually help the two students pass the exams with good marks all 3 years. It was not until mid 1997 when he was actually at Obed Siddiqui's lab in Tata Institute of Fundamental Research as a summer trainee that he came to know of a problem called dyslexia. You see, he was very relieved to know that he was not dumb. His problem was that back in 80s when there was no internet, you as a kid were respected, regarded, thought of as decent and well behaved if you could read. There was nobody who you could go and say, look, I can't read. The de facto response would be, you don't pay attention, you're distracted, you pay too much focus on cricket, your mind's wandering all over the place when you're trying to read. Focus, you'll be able to read.
Extremely sorry to disturb you, but maybe we can adjust the camera because on screen it's from below.
Yeah, now can you? How's that?
Maybe we can stand a little back.
That's all right. Okay. So at that time it was actually a taboo for anybody to go and say I can't read. That just means you're not fit to be a student. So the boy never went out, never told anybody he couldn't read. He was just trying his ways out to figure out and find ways to read. What took others an hour took him 4, but he had to do it some way, and it went on. So anyway, back to the main story. This guy went on to do his master's from University of Hyderabad, finished his master's there in biotechnology and joined CCMB for a PhD under Dr. Shivaji. And for those of you who are still struggling to figure out who that is, that was me. So now I got here. That was back in 1998. Now I got here. Now what? There's a very famous book written in 2007 by Marshall Goldsmith titled "What Got You Here Won't Get You There." The title was very appropriate for my situation back in '98. I got here because I had help. I had people working with me in teams. There were people that could read and I could comprehend what they were reading out to me. I had live help through these people. But then once I started my PhD, I realized I had to read on my own. I had to come up with my own projects. I wouldn't have somebody reading things for me. And I'm destined to fail if I stayed the course. It was just a matter of time. So I had to do something, and I had to do something fast because time was running out. So 6 months into my PhD program, I started doing, I gave a national entrance examination, qualified for an MBA course, and I started doing my MBA. And 2003, 1 year before I submitted my thesis, I completed my MBA. My goal was actually to finish my MBA and PhD, join, Reddy Labs, Shanta Biotech, some of those kind of companies those days, and transition out of active research, because science was still my passion. I had the broader umbrella of science as my passion. I wanted to be working in the field of science, but not necessarily active science, reading papers and writing grants. I knew that wasn't my forte. Um, you know, people say marriages are made in heaven, but for at least some of the students who join CCMB, CCMB is that heaven. Here's your degree, here's your spouse as a bonus. So, I met Roshni. Roshni was Ashok Kar's student. We were batchmates. And I must say, all the support that I've been talking about where, people helped me read, Roshni was that hidden support. A lot of people probably didn't know. I would have flunked in my coursework if it wasn't for Roshni. Roshni was my live on-demand podcast for my MBA classes. I used to be on call and I used to say, look, I have an exam in a couple of weeks, so you need to spend more time. So you used to say, she used to read things out aloud for me. Those are certain things that, they're hard to find. So, I was actually very fortunate to have had very good friends all along that helped me. So Roshni was that support even before we got married, or maybe that's why I married her, it went on, and after finishing my PhD, we got married, and Roshni wanted to go abroad to do a PhD, to do some postdoctoral research, and she was very enthusiastic about science. And I wanted to go to Reddy Labs and get a job, move out of active research. So we had this battle going on. Any guesses who won? Yeah, there you go. So as a side note, they say it's all about the war. It's okay to lose battles. I have been losing battles for the past 2 decades in the hope of winning the war. I'll let you guys know how that went when that happens. So anyway, I went on to follow her. She went there to Scripps in San Diego for a postdoctoral position, and I followed her. And the best and the easiest option for someone going from here to any part of the world would be to get a good postdoctoral position, especially if you're a graduate from CCMB. You practically are on par with any other research institute. You wouldn't need to relearn any process. It's exactly like what it is done here. Everything that you use is what you would use there. So you're a hot candidate for postdoctoral positions, and I got one. That was not my primary choice, but then I had to get some position to get somewhere. It kind of made sense at the time because rather than just jump guns and say I want to go to the industry and do something different, I needed to understand the work culture there. And postdocs are forgiven for a lot of things they do because everybody knows they are transitioning, especially if you're an international postdoc. So it made complete sense for me to take up a postdoctoral position. I got one at Sanford Burnham, and the process continued in my mind. I constantly kept on thinking that active research is not for me. I need to do something else. Now, how do I go about doing it? So I took up a project management course while I was doing my postdoctoral research. So I got a certificate in a diploma in project management, specifically geared towards drug discovery and development, taking the compound from, say, bench to bedside, all the way through the process of clinical trials, just to get an understanding of how the whole process is done, and also to have the understanding of the project management process for that. I'm hoping that that would be one of the paths that I could potentially take. So I was always constantly thinking of ways to, say, come out of active research but still be associated with science and do something more productive. And towards the later part of my postdoctoral research, I went on to work in the technology transfer office for 2 years. Again, something that complements the process of drug discovery and development. I was looking at patents, I was looking at technology licensing, and how agreements are drafted, and what's the process in the whole thing. I spent 2 years there, outstanding experience. I got to learn to also almost like a 2-year degree, working in the field. And this went on for a little longer than I anticipated. Again, I had to lose one battle to win the war. 2.5, 3 years into her postdoc, 2.5 actually, Roshni said, what, I don't think active research is for me. I need to balance my life. I remember someone was asking a question. I think Manjula was asking a question about how you balance your career with your family. I think Roshni is a good example for that. She made a choice. She said, I still want to be in science, but I don't want to be in active research. I don't think I can go 11 o'clock at night to take care of my cells if I need to do it. So she moved out to science, and I had to actually hold the fort, so to speak, while she was making the transition. And that went on for a couple of hours, sorry, a couple of years, I wish, 2 hours, for a couple of years. And then it was my turn to actually make transition out of academia. And based on what I was gearing towards, I started applying for positions in business development and product management and project management. So one of the positions I applied for was at Illumina for a product manager role. I went through the whole process of interview, and after the final round, the HR, his name was Sean, he called me and he said, unanimously, the entire team that interviewed you was of the impression that you're a better fit for sales than you are for product management. Would you like to interview for a sales role? I said, what the hell is sales? How do you sell? I had no clue what sales was. All I knew was those guys, the vendors coming, holding a bag and, walking around trying to sell stuff. I had absolutely no clue. But then I had to make transition somewhere. Then, as soon as I hung up the call, opened Google and said, how do you sell? So it was a fun experience, and I went through the interview process. Despite me not having any experience, they somehow thought that I'm a good fit for sales, and they said, yeah, you're hired. Those 2 years, I think, was by far, if I look back, revolutionary in my thought process. The way I used to think before I got my sales job for 2 years, and it was just night and day. You think in terms of time, you think in terms of deadlines more strictly than you did before. You tend to keep emotions out of your decisions more often than you did before. You tend to do more favors without these kind of deadlines because ठीक है यार चलेगा time है बाद में कर लेंगे. Sales doesn't work that way. I still remember the day when the director of sales called me, pulled me into a room. I was talking to a scientist, on phone. He was a metagenomic scientist trying to do some studies in soil samples using MySeq. MySeq was a new instrument those days. I was very excited. I'm from Shivaji's lab. I have publications on 16 sequencing, and this was a near and dear subject for me. So we went on for 45 minutes to an hour. As soon as the call was done, he called me into his room. I said, you know what you just did? You wasted 1 hour's worth of company's money. You're here to make money. You should know when to say no. This is too early of a lead for you. You're not getting business from him. Move on. Don't do this ever. It took me a week actually to digest the concept that you don't want to engage in conversation for that long if there's no outcome. What's in it for me? That was never the mindset before. I was always open for discussions. Going out, hanging out, having tea, fun. Very different mindset. So from that point on, I started looking at things in two different ways, depending on whether it is a fun project where I don't mind hanging out with somebody and spending hours, or whether it is something that I had to have a cut and dry deadlines. Nice experience to understand where to apply what. I still had something missing in me. I wasn't satisfied just working in sales. I said, I need to do something more. This was not just it. So while I was working at Illumina in sales, I actually went to college again to UCSD in the evening classes, and I got a diploma in molecular diagnostics from initially developing molecular diagnostics all the way through to commercializing the product. With all this as a package, I was fortunate to have gotten a job in a company, a small molecular diagnostic company called Pathway Genomics. So I took all my learning from this step and moved on to the next bigger thing, where I actually had the opportunity to set up 3 divisions on the commercial side, starting from defining the departments to hiring folks, to training them, to firing them if I made a mistake in hiring the right candidate. Great experience, and the toughest part was to fire people. It's always hard to do that, but again, I had to learn how to do it. There's a way to do it, and I got there. There was still something missing in the whole process, and I still wanted to do more. And while I was actually working at Pathway Genomics, my skills to actually go out, market something, position something in the space was lacking. So I went on again. I went to UCSD, took a diploma in marketing, and this was during the whole process, I was not really sure exactly what I was going to eventually do, but that gap always existed. I said, this is not cutting it. I needed to do something more, something bigger. And the quest continued. And in 2016, I landed up co-founding Triangulum Biopharma with 2 other people. That is again a very interesting story. But long story short, I've been interacting with somebody for almost a decade. And again, Roshni plays a role in the process, every step of my life. So Roshni, like I said, went to a job and she was working in a company called, those days, ISIS Biopharma, but now ISIS Pharmaceuticals, now it's called Ionis for obvious reasons. And I used to go to the annual parties every year, and we used to get drunk, late at night. And I used to meet one guy called Sanjay Pandey. And every year, we used to look forward to meeting each other, and we used to get drunk, and so much so that our wives actually used to drive us back home. And 2 or 3 years after, I started having a conversation with him, and one day I used to call him Pandey ji. Those days, now he's more like pals. I call him Sanjay. I used to have conversations with him and tell Pandey ji, आप क्या कर रहे हैं? आपका इतना अच्छा science knowledge है, आप किसी के लिए क्यों काम कर रहे हैं? Let's do something bigger. Company खोलते हैं. You walk the science and I talk the science. How about we make a company out of it, and why don't we create something bigger? He's a very conservative guy. Initially, he was very reluctant to the whole idea of leaving a good, comfortable job and forming a company. But eventually, he said yes, let's do it. Now I started thinking, how do I go about doing it? Because I said yes, but that meant leaving a well-paid job, and that meant starting something de novo. It was very scary. We didn't know where it would go, and I was projecting more confidence than I had when we were in the process of thinking through. It kept me up at night for sure, because failing was never an option. We just had to find a way to make it work, and that was beyond discussion. Again, Roshni comes to my rescue. We have had I had to group myself to put myself together, gain the courage to go. Have a discussion with her and say, look, I am going to quit my job and I'm going to start something, a new company, something different. She said, okay. And then I said, that means we need to take a loan on our primary residence because, one, we need to support ourselves for who knows how long, and we needed a little bit of money for buffer. She risked the roof on her head and said yes. She didn't blink an eye. Not just that, when I started the company and when I was working 16 hours a day trying to set up things for weeks, she took complete responsibility of of the house, the family, everything. Not a single word. It went on for weeks, and now she got used to it, so she doesn't complain anymore. From that point on, the story was good. We spent a couple of years, working on the company, and things went on really smoothly. We got very fortunate. Our strategies worked really well. They were in line with what we thought it would be. We are one of the very unusual sort of a company that we set up. We are a contract research organization, and typically when Indians form a company outside, Indians, Chinese, people from Singapore, South Korea, when they form a company in the US, they actually get business from the US, outsource it to India to a team that they actually build here, because it makes sense, because it's cheaper from a labor perspective, and they have more control in India. They can get things done the way they want. We had a completely different way of looking at it. We said, what, let's have everything in the US. There's a difference between chemistry and biology. Chemistry, it's cut and dry. If you have a compound, you say it's 95% pure, it is. You show it by mass spec. You ship it, they redo the mass spec. They know exactly what it is, what the purity is, black or white. Biology is different. You take the compound, doing a pharmacology study, you put it in an animal, how the animals were brought to the facility, all the way through to how they were sacrificed and how the necropsy was done. Every step is important. And showing is believing. So, we had the facility there. It was all, to the highest standards of regulation for international approvals. Everybody we had a conversation with was super excited to actually see a facility there. And, we've never looked back after that. And we've had fantastic business we've had. So we were recognized as one of the top upcoming companies in biotech in town back in 2018. We went on to help 3 different companies that were acquired, the most prominent one being Jecure, that was acquired by Genentech. And our data was a part of the presentation for the acquisitions. We've also had the privilege of working with NASA, and we worked on some of their space samples, very precious samples that they had, animals taken to space, trying to understand the effects of the ISS on liver. So we've had a wonderful run. We still continue to have very good success there. Things were still not complete. I still had something missing, and the missing puzzle there was, it's great, the company's doing well, but it's a contract research organization. I wanted to create a product. I wanted to at least give it a shot, because one thing I believe in is when you're on your deathbed, you shouldn't have a regret of not trying something. You should look back and say, I tried. I tried and I gave my best. If it didn't work out, so be it. But trying is everything. So I was thinking of how to come up with a concept to say form a company or bring up an idea for a project that would eventually lead to a product. And that's where my training with Shivaji in his lab in molecular biology and my experience in B-cell immunology came together. So I put both of them together. I worked on B-cell antibody production and class switch and class switch recombination and antibody maturation, so on and so forth. So all that training actually culminated to the formation of the company that's actually now incubating here in AIC. Called Mapgenics. We have 6 people, and coincidentally, well, not so much of a coincidence, we have hired 2 students from CCMB. I'm very happy. So that makes it 3 of us. 50% of the company actually is CCMB alumni. It's a great feel to actually have that sort of a setup here where you don't have to blink your eye. You can just you're going to get talent if you start something here. Absolutely fantastic. The way this place trains people, I'm always awestruck the whole process. So I would not say my life was all rosy. Childhood was actually pretty grueling, depressing at times actually, because I just couldn't read, and there was nobody I could share that feeling with, because we branded as one of those useless guys. It went on for 25 years knowingly, and maybe another 15 before that unknowingly. But I would say that if that wasn't the case, I wouldn't have started thinking different. I wouldn't have started thinking that I need more than just my path that is conventionally taken by most people to succeed. So that sort of helped me train my brain the way I think now. For me, what's happening now is less important than what should happen, say, 2, 3, or 4 years from now, because I feel that what's happening now is sort of set in line, and it's on track, and it will happen no matter what. Unless you have to do some sort of a damage control, things are going to go on. But the bigger question is, what are you going to do, say, 3, 5 years from now? So that sort of a planning mindset was de facto for me as time went on. And that probably was critically important for me, because everything I was doing, there was what next in my mind, and it still continues, because that's basically how I now start. That's the way my brain is trained now. And whenever people either speak about their experience in their career, or when they're asked to have some conversation with somebody about career advice, people always talk about passion. Very few, as I see, are lucky enough to know what their passion is young in their life, and they just follow their passion, their dreams. They succeed. PMB is a great example. When I was just having a few slides to show that there's reason why he's PMB and why he's there. Not very many are like that. But then for the rest of us, at least for people like me, I had to find my passion. I had to find my true north. And in the process, I just kept going. And the way I got to where I got to was, using the classic technique that when people give their competitive exams, you have multiple choice questions. and you may not know the answer to it, but you know one or two of them are not right answers. So what you do is you eliminate those, and you increase your odds of success. I started doing the same thing in my life. I knew exactly what I couldn't do because those were my weaknesses. Tell me where I'm going to die, I'm not going to go there. So I eliminated those as options from my process and started working towards trying out the other options which I thought were better fit for me. And that's how the whole process progressed for me throughout my life. And it still continues to be the same as time goes on. I am not by any means trying to glorify my success here. I'm not suggesting that I'm the best. There have been people much better than me. And there are people who are working hard and will eventually land up being better than me. It's just a mere attempt to show what my path was like. And if somebody could take a leaf out of this approach and succeed, I think my job is done for the half or 40 minutes that I'm going to have a conversation with you all. Nothing more than that. In the in the whole process, if I can summarize 5 key takeaways that I now would keep in mind if I were to do it based on my experience, and hopefully those will be good points for you all to keep in mind too when you plan your career in future. First thing is plan. There's a saying where they say if you fail to plan, you plan to fail. Plan the heck out of yourself. Planning is wonderful. It's like a game. The more you play planning, the more you enjoy planning. Because if you think about it, planning is nothing but dreaming with a deadline. Who doesn't like dreaming? Anybody here? Everybody loves to dream. So you just have to put your dreams, set them as goals and give them deadlines and work towards them. Not working towards it is failure, as I said, but if you work towards it, you fail, something went wrong, you switch. And it gives you a good feeling because now you're the decision maker in the whole process. And it gives you a lot of confidence in the whole process. Anything, as I see it, is done twice. Once in your mind and once in reality. And as I see it, when you start thinking about it, your brain starts building neuronal connections in that direction. So you actually have already started doing it. So when you physically start doing it, your brain's already trained to doing it because you thought through it, you know what to do. So that's point number 1. Point number 2, I think Rana is a great friend, by the way. I spent a lot of my time, needless to say, in South Wing ground floor. And Rana was Roshni's immediate senior. We were very good friends. We spent a lot of time together. I still remember, Rana, if you're listening to this, I still remember the days when we all went to Goa as a team. 6 of us went there. We had a wonderful time, in the Goa guest house. Great place. Anyway, so, Rana was mentioning, something about networking. I would say network the heck out of yourself, speak with people. Because I think there are 3 broad categories of learning. That there's one way of looking at learning. One is you learn from others' mistakes. One is you learn from your own mistakes. The third category, you never learn. All of us have, instances and situations where you can classify your experience into one of these. The more you skew the bell-shaped curve towards the learning from others' mistakes, the better you succeed, and the quicker you succeed. So have conversations with people. Just find people. Just have conversations with them. You'll know what they've done. You'll know if what they've done is right for you or not. How could you do that differently? Have that inquisitiveness to understand. Have a conversation with Vinay. Just knock his door. Go on. Sorry, I'm going to give you more work. But just have a conversation with him and see what what he could have done differently. Very similar to what questions you had with Rana. And this is a great dais for that, actually. The fact that you're listening to students and having some input from them is great. But I think it should go beyond that. It could be anybody start at the institute and beyond. That's one thing I would highly recommend. Because that's I think critically important. Third thing is teams. You can never do it alone. Starting with the days of my bachelor's, when I had people helping me read, I wouldn't be here without those 2 students. They may think that I helped them enough to pass, and they may think that they're eternally thankful to me, but it's quite the opposite. In the process of me trying to help them, they helped me more, and I benefited more from the process, and I found a way to live, found a way to survive, found the solutions to my problem. I don't know if and when I would be able to find a solution like that without them. So find teams, find ways to have. Rosh, for instance, is a great team member for me when I started my PhD. I wouldn't have finished a lot of things. So find your teams, find your teams, have those team player mindset. Just try and help selflessly. Be that approachable person that you would like to meet. If you're in need of something, you go somebody. If you're warmly welcomed solutions, think about how your levels kick in. Be that person to someone else. So always try to be a team player. So that's 2 to 5. That I would think in one bucket. But I think, in my opinion, the other two are critically important, and they are what decide how to use these three to your success. One of them is finding your true north. You see, there is this magnetic north for the Earth, and you also have what's called as north for your Earth. Magnetic north is your status quo. On a path, you just move on, comes your way. You say, yeah, this is next big. Yeah, take it. Yeah, not be thinking. It is great, but more often than not, it's not. So you're inherently, as a human, biased towards taking that conventional standard path and finding ways to just survive. It takes a lot of energy to deviate from the magnetic field and find your true north. It's a constant process. It took me a lot of time. It took me years. I still continue to do that. And it is exhausting. But I think finding your true north is what actually gives satisfaction. I would highly recommend it. Question yourself every time you're doing something. Jobs once made a fantastic statement. He said, you're looking at mirror in the morning and you're not excited to go to work. It happens continuously for a few days, weeks. You must question what you're doing. Again, that's having on the point that you need to find your true north. Levels don't kick in. Don't feel motivated to achieve something if you're just doing one of those run-of-the-mill career tracks, at least for most of us. The final last thing, but I think it's the most critically important thing as I see it in life, and again, Rana was asked this question, it's trying to understand and realize that career is a part of your life, at least for most of us. Career is not your life. So try and see how you would want to fit in your career into the bigger scheme of things. You don't know what you don't know. You've not seen the future. Most of us are not married. Most of you are not married here in the crowd, I assume. So when you're looking at it, when you're looking at your future, when you're thinking, oh, what do I do next? How do you fit in your life? How do you fit in your career with your life? You may want to be somewhere in your career, and then suddenly you realize 5 or 6 years down the lane when you have kids. When you're having to work with a working spouse, both of you working, 6, 8, 10, 12 hours a day. How do you manage that? Where do you see you fitting? Where do you see your career fitting into the bigger picture? It's critically important. I think at the end of the day, it's a complete circle. You need to ensure that you fulfill every aspect of your life and have that balance in your life. So don't just focus on your career. Always keep it in perspective with what your life is and where you actually ultimately want to be, and don't miss out of anything. When you look back, you should say, oh, I wish I did this in a more balanced fashion. That typically happens. So try and find your true north. It should be a process, and question yourself every so often, whatever is appropriate for each one of you, and constantly keep working towards that true north. It takes effort, because it's like exercise. It's easy to say I'm motivated, do it for 3 months, and then suddenly you realize, at least I do that, that I stop working out and have a big beer belly. Try and see if you can avoid that magnetic north. Try and find your true north. And I think success is just a matter of time. People will be there to help you. People always like to work with people who are productive. People always like people who are likable. If people like you, they listen to you. But if they trust you, they would look forward to working with you. Build that trust with people, and it just doesn't happen with one conversation. One conversation creates awareness. That's not networking. There's a misconception about networking. People say I have 2,000 connections on LinkedIn. It just doesn't mean anything. That's your digital network. But your true network is the number on your cell that you would call and say I need help, and the person wouldn't hesitate to open himself or herself to come and help you, show options to you, or have a discussion with you. That's your true network. Try and build a solid base of friends that you can actually work with and that are more than happy to help you, because they know. Keep in mind, when someone's trying to help you, they're putting their name at risk, assuming they're making an introduction about you or giving you a recommendation for a job. They're putting their name at stake. They should wholeheartedly be willing to do that for you, because they know you're going to keep up his word and actually make them feel or look good, because the other person would come back and say, oh, what a recommendation, that was just outstanding, I'm so happy. That's the sort of a feedback that they would love to see. So try and be that person. Try and convince your network that you're actually capable of doing something like that. So opportunities will flood your way. Trust me, everything else will fall in place. That's all you need to do. With that, I'd actually like to thank the organizers for considering me as one of the students worth listening to. I truly appreciate this opportunity. It's been a true pleasure, both from a memory recall perspective, walking down the campus and seeing some of the familiar faces around, and the warm response I got from the few that are left working here from 25 years ago. Great feeling, great pleasure. Thank you.
I shall.
Hello. Yeah. I request audience questions if there are any.
Hi, Suresh. It was a very nice talk. So you mentioned that MBA when you were doing PhD. I want to understand what was your thought process, why you decided to go doing MBA, and how did you manage it?
Second part was easy. Managing it was not difficult, because if you look at conventional MBAs, you take about 2 years. I took 4, because my goal was to educate myself. I was not meeting the 2-year deadline, so I could space it out. It wasn't that challenging, and I'm sure all of you will agree. If you intend to, you have enough time during your PhD where you can do something else more productive. The thought process was, again, it falls in line with how things were for me throughout my life until that point, where I knew what I was not good at. I knew what was not going to be an option for me, and I needed to find ways to supplement what I have so I could get to a better place. If you think about it, until then I was in a survival mode. All along, my goal was just to survive, just to ensure that I stay put and not fall from where I was. That was not enough. I actually need to do more. In the whole process, I just need to keep building it. And MBA, because people who, if you see some of the old folks, they'll tell you I was notorious around. I was very vocal. I was a chatterbox, as they used to call me those days. And so I had this other side of me. People, in fact, Shivaji once, I remember when he came to know that I was in the process of doing an MBA, he said, "You're in the wrong place. You should not be here. The way you are and the kind of training you're getting, you should be somewhere else." He was spot on in identifying that. I was not meant to be in working in a lab on a bench. I felt very claustrophobic, honestly, working in that small lab, wearing a headphone and just keeping to myself. It's very difficult for me to do that. I am not that by nature. But then I knew not having a PhD, is not good, and giving up was never an option for me. It still is not. I generally try to fight it out to the end, and it is very depressing if I have to give up something. So it's hard. So that was never an option. So I need to do something else, and that's how the whole. I had thought of a lot of things at that time. Nothing came to mind that was more convincing than having an MBA. In fact, my MBA was in general administration, operations research and operational administration, because again, putting all things together, my background is I've done my bachelor's in microbiology and genetics, and my master's was in bio. I was working in a lab where I was doing microbiology. So if you look at it, everything was adding up. So if I say went to radio labs and actually worked in a say fermentation department and doing some organizational operational management, that would make more sense. So I was trying to build up based on what I have and how I could potentially use it to take it to the next level. So that's how the whole thought process came, and that's where my specialization, my MBA, was also dictated. Hence the marketing diploma later point, because I only had a smaller base. It was 20 odd years. It's always nice to refresh your knowledge. So that's how the MBA idea sort of came up, and that's how I worked towards it.
Any questions from the online audiences?
Suresh, can you tell us briefly about what MapTech is about and what it's doing now?
Oh, Mapgenics, yeah. Yeah. So Mapgenics is actually an immunization-free antibody production company. Again, aim high, right? So I don't want Mapgenics to be one of those conventional service providers. The aim there is actually to try and come up with a product, take it to the market, global market preferably. Mapgenics actually has, we've developed a system where we've mimicked the immune system that could actually reproduce all the permutations and combinations of somatic hypermutation and potentially close to represent the actual diversity of antibodies in the system. And like I said, it's a combination of my training here at CCMB and what I've learned during my postdoctoral time when I was spending in Robert Ricketts' lab at Sanford Burnham. We are in the process of building the platform. We hope that will be done in the next 6 odd months. We see no reason for the platform not to work because of the way it's designed. Now, how diverse is the platform is what's going to define how well the platform will perform. But I don't see the platform not functioning. With that in mind, the ultimate goal is actually to work towards therapeutics. But I'm very realistic about things. So we are actually going to have multiple attempts in the process. We're going to work on 3 or 4 different areas, most of them simultaneously. We're going to hire people to work on each area. We've already hired 2 people working on 2 areas. We're going to be into biosensing, where we're going to develop antibody-based biosensors. We're going to be in diagnostics. And the primary kind of antibodies that we're working are nanobodies. I'm sure most of you, if not all of you, have heard about it. Great molecules to work with. We have a lot of advantages. We actually want to take those advantages and see if we can build something around that. So that's where the company is. It'll shape up as time goes on. It'll start settling down, go through the funnel, and finally come up with 1 or 2 key areas of research for the company. But that's where we are as a company.
I'm a long time user of nanobodies. I kind of use it in the lab all the time. So since you are saying that it's a therapeutic application, Like I was thinking that there are a lot of academic institutions. They use the a lot of antibodies. I do see a transition from antibodies to the nanobodies. Are you considering the research institutes also, because India is a big market, I would say, for that. Like, they're able to provide a nanobody-based antibody business, because now since India is promoting this Atmanirbhar and all those concepts, so I'm just thinking, do you have that also in your mind? Because we have so much institutes and the universities in India.
So I've already had prefix to that. We don't, at least at this stage, intend to be a service provider. Again, there are investors in the company. At the end of the day, you never know what shape it's going to take eventually, but that's not the intent with which the company started. In terms of collaborations, I've already had conversations with 2 scientists at CCMB, and we have potential ways of working projects out. So it's going to be an academia industry collaboration, to build something in that direction. Certainly, one key to success is to find ways to complement your skill. We are not experts in certain areas, for instance, cardiovascular disease. We only know the technical aspects of how we can come about doing, say, raising an antibody or optimizing it, so on and so forth. We are not CVD experts by any means. So what we do is we actually have partnerships with people who've been lifetime CVD experts in research. Same is the case with biosensors. We're in the process of building and optimizing biosensors, but we are no way engineers. We actually have a very well-renowned, scientist, Dr. Sudesh Sukla, on the board, who's an advisor, academician again. So we very strongly believe in leveraging the talent pool in India. And that's one thing that I'm really focusing on. We're trying and avoiding as much as we can to have people who are outside India on the board of the company. Some cases it's not realistic. Some cases you just have to do it. It makes sense. But academic collaborations, yes, are on top of the mind, but not as some company providing antibodies for academicians to use, more as a co-development method to get the whole thing to the market. I'm sorry? Yes, looking forward to that. Yes, thank you.
Hi, Suresh. Nice journey that you have taken us. So my question is, when we see around in the globe, when the scientists which are doing the academia, also they are having the industry, they are also setting up the industries and all those things. So what do you think, what kind of scenario will be there in India, if people who are in academia and the side by side they also want to set up a startups and all those things? Do you think where are we going in that direction?
That's a great question, actually. I'll comment based on my observation and some of the discussions that I've had with a few of the scientists around. What I have seen in terms of companies spun out from academia is purely on the basis of the merit of science. More often than not, what lands up happening is that you may have an outstanding idea, great product that has no market. If you can't sell it, there's no way, your company is going to succeed or your idea is going to see the light of the day. So from an academic perspective, I see why that process actually comes up, because you think of a project, you have a hypothesis, it's going to work, you go about doing it, you aim for nature, worst case you publish in JBC, you're eventually going to have a publication no matter what. That doesn't work on the commercial side. You're aiming for nature, it has to be a nature. So if you think that it has market potential, if you can't see the product in the market 5 years after you launch the product, then you're working on the wrong product. Because it's not going to give you the sort of revenues that you would need to see for that to be a viable option in the market. We discuss a lot of projects internally, and 1 in 10 would actually make the cut. And think about the challenge from that point on to actually make it a viable project from a scientific perspective. So we start thinking, at least I start thinking in just a flip way, with the whole process. Identify the market where there's a need and see if there can be a product there and come up with that. So that's a sort of a gap I see in the thought process for people coming from academia and trying to spin out companies. It's a great process to do, hands down. Uh, Science is born in these kind of institutes. Spin-offs are the way to go. That's how billions of dollars are made in the international market from various places in the world. There's no denying that. But then that's one thing I would suggest be kept in mind. How are you going to end up? Where are you going to see this? Fit in the broader scheme of things. How do you compete with the existing technology? Great example is metformin. There's a reason why type 2 diabetes research is actually on the decline. Not many people actually work in the area because metformin, get a pill for like I don't know, 5 rupees, 10 rupees, and you just keep taking it for your life and you're old. It's hard to compete with those drugs in the market. Outside that market, come up with a drug, with investing $800 billion into the drug, what's ROI, and when you're going to actualize, realize profits. So that's the challenge in mind. So the only thing I would do if I was that academician is to have a conversation with somebody in the industry or start thinking in the big way, start thinking with the end in mind, rather than saying, well, this is a great product, where do I fit in? I think that is less succeeding in the broader scope.
I suppose there are no more questions, and thank you so much for the session. And I would request Dr. to begin.
Thank you. Thank you. Thank you.
అర్థం కొడుతుంది फोन को वही बोला आते ही लैपटॉप निकाला ना बोल तो अन्ना इसका सोच बैठ हुए थे इसको भी
इश्यूज लाने के लिए
वो तो ना एडे పెడతా అన్న వాటర్ క్లాస్ తీసుకొచ్చి మరి
कुछ कपड़े तो दे दो बोलो ना नीचे से
विषय पట్టుకొని
Hello. So I again request the audiences to take their seats. We are going to start with student talks. We are going to start with the student talks, and next up I'm going to call upon Priyanka, who is going to speak upon the non-coding codes. Priyanka, the stage is yours.
So, hello everyone. So, I'm audible? Yeah. So, hello everyone. I am Priyanka, and as many of you know that I work on long-coding RNAs for my PhD work. And today I'm going to talk about, well, few link RNAs which are actually coding. So they are not really non. So let's start. I'll take you back to 1st October 1990. So this was the time when Human Genome Project was officially first started. And so the idea was it was funded by this Department of Energy, the US government. And the idea was to do the whole genetic blueprint in order to study the DNA structure, damage, repair, and how to really clear it. So remember now, World War happened 45 years ago, and there were a lot of mutations coming up in the population which were widely studied. So that's why Human Genome Project was first started. That's how it was funded. So about a decade later, we had the first draft of Human Genome Project, and now the biggest task was to assign meaning to these bases, which we got about 3 million bases, 3 billion bases, yeah. So the process of annotation started. Now, even before we started sequencing and the first draft was released, we already had protein databases such as Atlas of Protein Sequence, which dates back to 1965, protein database started in 1971, and then SwissProt, which started relatively recently. Now, keeping these as a initial, platform to build on, the GenBank and EMBL DNA library, they started annotating the sequence. So this was totally protein-centric way to annotate our genome. So the chromosome 21 was the smallest one, and it was released in 1999. And keeping this in mind, two groups independently, they did a microarray, and what they are finding, like they took a tiling array of probes and placed it on a DNA chip, and then they studied the transcriptional noise coming from chromosome 21 and 22. So what they are finding, that this is a chromosome sequence as generated by Human Genome Project, and these are the annotated regions. So there was a lot of transcriptional noise apart from the gene-rich or annotated regions. So now this was the starting point when people started questioning that there is so much more which is transcribed apart from the protein coding region of the genome. So there were a lot of microarray data being generated, and there was a need to compile all this into a repository and in an accessible manner so anyone can access it. So this is how ENCODE was born. So ENCODE, or Encyclopedia of DNA Elements, it aims at cataloging all the functional DNA elements, whether it's protein coding region or non-coding regions. Like it wants to, the aim of the whole project is to define everything about DNA, and it's still ongoing. It's presently in the phase 4. So in my talk, I'll focus on the long non-coding part. So these non-coding RNA, how do we define long non-coding RNA? So these are RNA which are greater than 200 nucleotide, and they do not encode for any protein. Now, when I say protein, there's a star mark, right? So these proteins, well, the definition was any peptide more than 100 amino acid. So this was the cutoff, arbitrary cutoff, which was decided. So if you see the coding potential, like this is GAPDH, for example, so you see all these big, huge ORFs. And if you compare this sequence with a non-coding sequence as ZIST or TUG1, you see the ORF size is comparatively small. And it was assumed that since these ORFs are very small, less than 100 amino acids, they might not have any, functional relevant. So how, when we, discover some new link RNA, how do people actually study it? So they first confirm whether it's non-coding or not. So they do the race, find out the full sequence, and then they do in vitro transcription and translation, run it on a page, and then see that there is no peptide band coming. Other way to do would be overexpress this link RNA, perform a mass spectrometry, and detect any of these predicted peptides. So now there was a flood of this transcriptomics, and a lot of non-coding RNAs were being studied. Many of them had actual functional relevance. But one group in 2009 asked this question, like how much of this transcription which is happening is actually getting translated? And they came up with this technique called as RiboSeq. So here you isolate polysomes, you do RNase digestion, and then you capture all the ribosome protected RNA fragments. So the idea here is whatever is protected by ribosome, is bound by ribosome, is actually getting translated. So when they actually check the translational efficiency, so here the black dots, this represents a few link RNA genes. they actually found that a lot of these annotated link RNA actually had comparable protein coding potential compared to protein coding genes. So they were actually annotated as link RNA, yet they had a coding potential. So the question now is this even physiologically relevant, or is just pure translational noise, right? So now we have like a lot of data being generated, transcriptomics is there, RiboSeq is there. So, people started to sort of overlap these transcriptomics data with RiboSeq data, and then the concept that if a peptide has to be, if the peptide is functional, it has to be conserved. Okay. So in 2015, one group from UT Southwestern, they discovered this small 46 amino acid peptide sequence called as myoregulin, encoded from a link RNA, like encoded from a transcript annotated as link RNA. And this peptide sequence, so we have these in skeletal muscle, especially this peptide sequence was mostly enriched in skeletal muscle, and in skeletal muscle, this SERCA channels, they are very important to maintain the calcium homeostasis. So what this peptide was doing, this was inhibiting the SERCA and causing the calcium increase in calcium in the cytoplasm. And when they made a knockout animal, they observed that these animals were sort of marathon runners. They abolishing this MLN function is causing them to run for a longer time and a longer distance. So this was very interesting, like even such small peptide can also have function. And after this paper, there were many papers which were talking about different peptides. So one of them, let's say this BNLN, it is actually decreasing calcium level and influence insulin secretion. Then OXY, it is regulating beta oxidation, and this toddler, it promotes cell movement during gastrulation in zebrafish. And if you abolish toddler, there is no gastrulation and the embryo development just stops. So far, these are the link RNAs which are actually misannotated as non-coding. So they were not studied ever before. But what about the non-coding RNAs which are already being studied? So I mentioned TUG1 in one of my previous slides. So this TUG1 is identified in 2005, and it was identified as a non-coding RNA required for differentiation of murine retina. So a recent paper which came in 2020, a group of researchers in Colorado, they developed a knockout mice for TUG1, and what they're observing that these mice develop male infertility, the sperm motility is decreased, and this being a non-coding RNA has a lot of cis-acting role. So in the TUG1 knockout mice, there was upregulation of the neighboring genes, which is very much like how non-coding RNA behaves. But what they find that they found that in the first exon of this transcript, there is a mini protein, which is of 153 amino acid long. This ORF is conserved, and when they overexpressed it, it was shown to be localized in nucleus and in mitochondria. And overexpression of this peptide is actually causing decrease in the mitochondrial membrane potential. So this is one example where a non-coding RNA is acting, has a both non-coding role and protein coding role. So this is one very interesting paper came in 2020, and here it was a collaborative study between multiple groups, and here what they did, they collected human hearts from almost 80 humans, and there was a control group, and they compared it with the dilated cardiac myopathy heart, and they did RNA sequencing, ribosequencing, and deep proteomics to understand the small proteins of heart and of iPSC derived cardiac myocytes. So what they observed that when you compare RiboSeq data with the proteomics data, indeed those non-coding RNAs are actually coding for small ORFs, which have an average length of around 46 amino acids. And interesting part was that this ribosomal occupancy is actually changing from the control heart to the hearts which are diseased. So here this dark red region is a set of link RNAs, which are actually encountering more ribosomes now in the disease condition. So again, pointing to the fact that whatever we were ignoring, considering small length, and we should really start focusing on, because maybe these transcripts are not coding after all. So they again selected few of these transcripts, and they did in vitro translation assay, and what they are finding that if you take the full sequence, they do produce a peptide, which is getting abolished in the delta ATG when you abolish the stop codon. So overall, we have seen that, there was a lot of this hype about non-coding genome, and now there are papers coming up that after all, these non-coding genomes might harbor a small peptides, which could be functionally relevant. But what is interesting, like, why do we have such small peptides? Why is there so much of translational noise? So there is this one paper which came in 2012. So this talks about that this SORF can be a sign of evolution happening. So when a new gene is formed, we know that there are non-genic sequence which can or genes can be duplicated and they form pseudogene, and they later on might acquire new function. But how do genes actually originate de novo? This group is proposing this protogene hypothesis, where they say non-genic sequence might acquire a status of protogene, where it might encounter RNA polymerase, it generates some transcript which might get translated. And if these transcripts have a minimal non-specific function or some weak specific function, eventually over the time they are selected, and the mutation in the stop codon might cause the gene size or the ORF size to become bigger. Now we have a lot of this data from RiboSeq, and people are annotating and reconsidering whether what kind of non-coding RNAs, like how non-coding are actually the non-coding RNAs are. So there is a need to build databases for such small peptides, and so few people from few labs from US and Europe are actually making some attempt to characterize these ORFs based on their position, or whether they are encoded from a non-coding RNA, and are actually trying to coalesce it into this UniProt database. So to summarize, Human Genome Project, it started in 1990s, and were roughly 20,000 protein coding genes were estimated, and the whole annotation was largely protein centric. So after this, there was a lot of research which say that there are actually non-coding regions also, the regions which do not code for any protein. And these non-coding transcripts, how they were defined was that if they have ORF, which are less than 100 amino acids, they were being ignored. But now, after RiboSeq, when people discovered that ribosome can actually bind to these non-coding transcripts and produce an active functional peptide, I think it's time to revisit those studies which we have done to actually find out how many of such ORFs we have ignored in the process of moving ahead. So yeah, these link RNAs, sometimes they can encode or harbor small ORFs and have a functional relevance. So it's not just protein coding or non-coding. There could be transcripts which might have both the kind of functions. So yeah, that's all. I'll be happy to take any questions. And
are you able to hear me, Priyanka? Can I ask a question? Hello?
A simple ORF, which is in hardly any function. And then after that, you have the ORFs which are increased, at least for me, there seems to be a roadblock about so having a low function to having a weak function.
so actually, so this study, they actually took around 10 yeast strains, and they are comparing few of these ORFs, and they are able to see that some of them have ATG, some of them don't, and hence there is a, some of them produce some small ORFs while the other strains do not. So it can happen. It could very much be probable, because if there is some region which has ability to encode a small protein which has some important function and is, you know, conferring advantage over the existing organism. So yeah, I think in future it can get selected. And in this paper, like I can share this paper with you and you can read more.
Nice. So my question is basically like, can we think that it is like kind of a new book? It may be the other way around, because we see that the evolution that has taken place. And we see smaller like theoretical, there the introns like non-coding is right. So all of a sudden when the eukaryotes came, so there is lot like this non-coding part is expanding and expanding and expanding. Why do we see as like it's kind of like whatever it can be like the lot of chunk that is being taken from the other organ, maybe like presenting transfer or like can be many. Maybe those whatever the proteins which were being encoded over there, they are now being fragmented, and that's a link with to get the new layers or new eukaryotic innovations which are being.
But again, when those sequences are integrated, you are trying to say that they still will retain the old function.
Yeah,
and
I'm saying that there is fragmenting.
Project is seen not selected. That project is not over.
Is it possible that protein coding gene is becoming non-coding? I'm sorry, I don't get your question.
You can help, like, can you send me there some screenshots from which we can, whether we will be able to make out that you are able to select with the screenshot, so that I will forward this. Send me email upon a monitor basis. I will try to figure it out. What is your contact number?
We have Sivakumar Prasad talking to us about the evolution of multicellularity.
Yeah. Something is. Your friend.
Good afternoon, everyone. So, as everyone aware, like nowadays, everyone is much more fascinated and interested about the single cell omics. Like you must be seeing, like there are like multiple different tissue types are there, and there are different single cell sequencing coming, and they are unraveling the multiple of the multiple biology. But if you see very carefully, everything came from a single cell. Okay, now I am trying to explain you how the single cell becoming multicellular and what is the advantage it has. And as we know, oxygen is the major driver of the evolution that many people know by the fact that it is necessary for the burning and like burning the fuel and so many other biological function. So now I will try to combine both of them and try to give an evolutionary perspective how oxygen would have helped in the multicellularity. But whatever you have thought, and I would say they will be wrong. So first we will define what is multicellularity. Multicellularity can be defined into two forms. It is my definition that there is something called complexity, there is something called aggregation. When you get a bacteria in a colony, that I would call an aggregation rather than a multicellular complexity. when you see this one, the zebrafish which is developing, it is coming from a single cell that it is developing into a multicellular complexity. But you see the sardines, they are kind of aggregating, even though they are doing a very important function that escaping from the sharks, but I would call them a multicellular aggregation. Okay, so this is just to keep the the concept clear that we are going to talk about the aggregation which is connected, not necessarily something which is dissolved. Okay, so for the connection, what we need is very important property of the molecule, that is sticking together. There are there is a research happened, it's a small molecular simulation experiment have been done with respect to the idea of molds, slime molds. They are the very good model system for studying multicellularity. As you can see here, the non-adhering cells, you can see there is a red color gradient and yellow color gradient. Red is the maximum substrate availability that is shifting over and over. And also you can see this is another there is a one semicircle is moving here, that you can see that there is a group of cells are moving across, and this is othering cells. As you see, the simulation clearly says that if the cells don't have an othering property, they don't form a multicellular aggregates. Okay, so neutrally othering cells can move very fast and they also make a multicellular aggregates. As you see here, othering cells, they are even though they are not able to utilize the energy very well, as you see, mostly they are centered. Okay, so adhesion is an important driver along with the resource availability, which drives the multicellularity. So in order for the multicellularity, first thing you need to get is adhesion. That's I want to emphasize. Again, what is the advantage of multicellular aggregation in the concept of they are sticking together? Again, I'm taking the sardine example, that the first thing for the evolution to happen is the escape from the phagotropic organisms. Phagotropic means something engulf. You can imagine, there is a small organism is there and there is a big multicellular organism is there. What happened to the mitochondria or chloroplast? It just engulf. Let's assume the mitochondria is trying to escape from this engulfing phenomena. Then they will start making an aggregation and they will try to escape from the bigger prey, bigger predator. Okay, so that was the it is one of the, I would say, nothing as the one thing which defines the evolution. It is probably one of the definition which defines the initial origination of coming together. And the next part is making the canvas ready. What I meant by making the canvas ready is, once you have the cells coming together, then all you need to do is, with respect to the environment and with respect to the selection pressure, you just start evolving and emerging new characters with respect to that. So that's what I make by the making the canvas ready for complexity. And it's not whatever I said wrong, because there is almost 25 times the multicellularity had emerged, disappeared. Okay, and the most successful one, as we see now, are three major ones are animals, fungi and plants. They undergone the multicellular complexity, as I say. There are other clades in the evolutionary timeline. You can see, especially the slime molds, they appeared 2 times independently. They have generated the multicellularity had emerged in that. So it is easy to get multicellularity. Then comes the oxygen. This is a very appropriate figure, I would say. I have put O2, O2 here. As you see, there is a gradient of what I would say is there is something called oxygen control hypothesis. So what it proposes is the body size corresponds to how much oxygen can diffuse inside. In the sense, let's say in the atmosphere, if the oxygen is less abundant, then what happens is there is so much can go inside the cells. If there is more oxygen, there is something more can enter inside the cells. So based on the idea of the oxygen penetration inside the multicellular cell, they propose that oxygen is the driver of multicellular innovations, and there are there is in fact evolutionary lot of evidences for that. There is something called the great oxidation event. In order for the simplicity, I have put one non-scaled graph over here. As you see, the great oxidation event, which happened around 2.3 billion years ago to 2.4 billion years ago, where the atmospheric oxygen started steadily increasing. Okay, so how anyone can say that oxygen status of 2.3 billion years ago? It is one of the important question, and the underlying phenomena of understanding how the oxygen is there itself is a lot of controversial statements. Basically, first controversial statement is that based on the geological predictions, they have found about the presence of the oxidizable elements and non-oxidable element in the Earth's different layers, and like aligning with them with the carbon dating information. Based on that, they will figure out how much was the oxygen amount and how much was the purely based on the amount of red soil on the earth crust, they have predicted that 2.3 billion years ago the oxygen started forming in the earth, and it was very well believed, and for the past 50 years there was no dispute over this. And even one more thing to add to this is the oxygen which is steadily increasing over here, and the size of the organism which is increasing. As you can see around the dinosaur emergence, the oxygen level was so high that they believed so much that oxygen is the one which is driving the multicellularity. But it is more of an analogy. There was not much of an experimental evidence. But now there is one new experiment people have done based on the a different approach. They were not checking the soil and saying that the oxygen is there or not. They were in fact checking the sulfur and also doing the mass spectrometry analysis on the different crust, and predicted that this particular cluster which is the red color mark, it's the sulfur data from their data set, which represents the high sulfur content. They measure, I can send you the reference, it's very geological terms, I don't want to bug you with that. So but the interpretation is, if you can find this, that means the atmospheric oxygen level should be close to zero. It should be 0.001% of atmospheric oxygen have to be there in order to get a meaningful data here. Okay, so that their point is not to dispute the oxygen formation, because in fact here you can see the increased oxygen even from their prediction. But what they are trying to say is oxygen did not started forming only during that time. It also started forming as a fluxus even before the period which is dedicated as the oxygen formed time. So the great oxidation event lacks oxygen. That's kind of now we need to question, is our multicellularity definition, is it correct or not, is the question which we need to ask. Well, there is already researchers asked. I will come to that part immediately soon. So summary is over from these discussions. Adhesion is necessary for multicellular emergence. The greatness of great oxidation event is in jeopardy now. So now I will tell about the experimental evolution of multicellularity. It's a very nice group from Georgia University, America. They are doing this multicellularity experiments. So what they do is they take the yeast and they mutate it to ACE2, because as you know that I already established that adhesion is very important for the multicellular innovation, that in order to get it, in order to do an experiment, you can make an ACE2 mutation which makes them forms a bud and they are attached to each other. The yeasts are kind of attached to each other as shown, and they are called snowflake yeast. That is the first thing they take, but they are not kind of a very big arrangement. Okay, now you take the snowflake yeast. In that snowflakey background, you make the mutate mitochondria. Someone can ask how you can mutate mitochondria. So mutating mitochondria means they just abolish the mitochondria's activity, which can oxidize, which can use oxygen for its energy making mechanism. And also yeast, majority of its carbon source is glucose, so they will supplement with the glycerol. So it have to only undergo the anaerobic respiration. So it is called strictly anaerobic, the condition 1. The condition 2 is strictly aerobic condition. So what they do is PAL means present available oxygen level, and 24% of that plus glycerol. That means it cannot use glucose as an energy source, and it have to use oxygen as an energy source. And the third one is the normal glucose, like a growing mixotropic snowflakey yeast, and it is also having the same level of oxygen and the ancestor for the control. Okay, as you say here, this is the probably one of the first multicellularity evolution experiment which have been performed, and it gives lot of insights. So the experiment, and I will ask some of your opinions about the like outcome of these experiments. Like I can ask you to raise hands if you can. Okay. So they have done this multicellularity evolution experiment based on your pre-assumption and based on previous ideas. What do you think will be the outcome of this experiment? Do you think who will make the multicellular form early, and like who will survive us and which will be growing as a multicellular organism? Like anyone for strictly aerobic, anyone can raise hand that aerobic will lead to the formation of the multicellularity innovation. Okay, so I think everyone is convinced with the anaerobic, with my arguments. So as the experiment, what they do is they take the multiple cells and they let it settle and they change the media, and this happened for 3,000 generation. As you can see here, this is the ancestor which was used, and this is the evolved one. You can see the chunks of cells. They like how they do is they just take the test tube, they just settle it for immediately, take the liquid out and take the pellet. It's not the centrifugation pellet they are doing. They just whatever the gravity settlement which happens, immediately they take, and like that they do the experiment for multiple generation. So and the winner is the anaerobes. So the ancestor was also like it's kind of like you can see the like it is the actual multicellular. But when you like actually see the strictly anaerobic category, they evolved into the multicellular form more. And as you see, after when they went ahead to the 3,000 generation, as you see, this is the anaerobes which makes the biggest size. Like as they have written in the paper, the yeast cell like evolved into so much. Now it is looking like not like an yeast anymore, which was looking like a gelatin, like not even less than the like tensile strength of a gelatin. Now it is looking more like an wood. Like they can even cut it with a scalpel or something. The size had increased almost as the size of a Drosophila. Now we can like think about like is it like really the oxygen was the driving force of evolution? Now like I would say we should not think in a singularity like that like one thing controls everything, like one gene controls everything, one concept controls everything. So we should be open to the idea that there are many factors could be there. One of the interesting factor is the nitrogen fixation. I'm just telling without slides that like nitrogen fixation cannot happen when there is an oxygen is there. So you can see the nitrogen fixation is kind of settled down to the roots, but when the photosynthesis which is uses the oxygen which settled up in the different areas, so that way you can separate, and the still oxygen is a driver of the complexity here. But is it the initial canvas made or not? That further experiment need to be done. And there are some like interesting for you the audience that embryo development inside the when it is developing, it is under the very low oxygen condition, even the bone morphogenesis, and in fact the myocyte differentiation in plants which I work on, that is also very much like hypoxia condition is necessary for the myocyte differentiation to happen, and also stem cell maintenance, especially for the tissue engineering. People have shown that like if you keep your culture in a very low oxygen condition, the stem cell maintained better, and like it is always compared to the surrounding cells. So it is also considered in the tissue engineering applications. And like I would like to kind of say this quote, I just quoting from a paper. Nevertheless, although the transition to simple multicellularity may be relatively easy, there appear to be less well understood obstacles to the evolution of multicellular complexity. And one more important question still remains is like when you have a multicellularity, yes, it's might and great, but how you are going to bring the memory of multicellularity to the next generation? Like during the like reproduction or whatever, just now aggregation is there. How the aggregation memory is actually going to the next generation? Like how I don't think anyone have asked this question like really carefully. I think we like there are multiple avenues are there. I end up here for the questions. Thank you.
It was a nice talk.
Yeah.
Looking at it in the context of localized tumorogenesis.
Yes.
Now, based on what you said, a localized decrease in oxygen induced.
Well,
but also the tumor to exist angiogenesis.
Yeah, like I was reading these papers with respect to tumor. I also reading, but in any case, like the mostly when you grow, like when the cells are growing in a multicellular complex organism, there is always something called defectors. Like the defector is the mutation which can, the somatic mutation which can happen in the cells. They can kind of like cause this kind of abnormality. Okay, that is true. Like they kind of have both the like. Yeah, true, true. Even angiogenesis, for that matter, like kind of happens in low oxygen condition. If you see the, I haven't put that image here, but the entire angiogenesis, the inside part of the where the differentiation have to happen, there it kept the lower oxygen level. So it is not a like what I am trying to explain from here is not a single oxygen presence or not. It's like how Ullas Koltur have given a talk recently. It's about how the gradient and what are the other factors affecting the cell's outcome. So I think like I can't really comment on the tumor and how it is evolving. We can discuss it. Yeah.
So when things aggregate, your cells aggregate with oxygen, do you see or observe certain patterns or is it a random aggregation?
Yes, very, very interesting question. Like I can bring up the analogy of a very nice this story. Like you can take a straw, like you can take a bundle of straw, and if you take one straw which we usually drink, and you can bend it easily. Okay, so and like if you keep the straw in a multiple of them, and you cannot even bend it. So the aggregation, whichever you are expecting, it have to be kind of a linear agreement, like how muscles are kind of stacked to each other, like muscle cells kind of. So the aggregation, not necessarily just the normal aggregation won't be enough. It have to be very strong aggregation for the evolution to act in the level of settlement and all those things. So even like I haven't elaborated in the multicellularity paper, they have shown that whatever the evolved cell, when they do a cut and do an experiment on that, they always make a elongated tube kind of structure. So that is necessary for the evolution to happen with respect to the multicellularity.
Yeah, great talk, sir. So my question is, since you've been saying that you ended with a quote that said that understanding the evolution of multicellular complexity is very difficult.
Yeah.
Is it possible that even for the existing multicellular organisms, it went through a stage similar to slime molds or dictyostelium, for example, where it's multicellular but not multicellular in its true essence? So is that a possibility?
I think that like that is kind of what I would say the first the canvas making ready. Like the initially the everything must have to be started from that. Even people suggest the animal evolution and fungi evolution to be little bit different, because in animals there is no cell wall and there is no attachment per se. Okay, so but there is a based on the ecological evidence and the carbon dating evidence, they have shown that initial ancient animal lineage, they do have the cell connection like how fungi and plant do have. So like it is possible that everything kind of starting from a similar pattern like what they are seeing. So you make the aggregation and give a different conditions, like you can get energy from one direction of the area, the niche which you are growing, and the other direction kind of excluded out. Then that cell become a different cell identity. So that kind of selection process can act on the multiple levels and can lead to the multicellular emergence with the complexity. Multicellular emergence with complexity. Yes. One more. Okay. Like, yeah. Pradeep.
Hi, Shivan.
Yeah.
Like, nice presentation. So my question is like, I'm not like against broccoli whatever you are trying to say. There are like some sense that you are trying to make out of whatever it is. So my question is regarding like if you are trying to say that in the case of plants also the multicellular evolved, and if you are saying that it is the anaerobic environment that has pushed towards the multicellularity, but the plants, how basically the emergence has like appeared when the cyanobacteria which came into the picture, and it is the one which provides the oxygen that whatever we have. So basically the cyanobacteria is the root cause of the oxygen in the environment, And also, if you
that root cause, I can't agree to that point. We can go ahead. Yeah.
But when it is inside the plant cell, when the like algae, all the algae where the cyanobacteria was there, it was already producing the oxygen inside. So in that plant multicellularity, how can you define that it was the anaerobic nature? Because it is already producing oxygen within inside.
Yes.
How can you make the environment, whereas the environment outside may be anaerobic, but still you have the aerobic inside?
Yes. I can understand the point which you are trying to make. But if you see, like as you correctly said, that cyanobacteria was the like initial push for the oxygenating the earth. That is true. But even then, like whatever you are saying about the engulfing of the mitochondria, like we can't be sure still that like I'm not saying that multicellularity as a what I'm talking about the multicellular complexity. When you talk about the multicellular complexity, there comes whatever you are saying. Okay, when as far as part of the single like aggregation of the cells to happen. Okay, when don't think about mitochondria or chloroplast being present already. Think about the scenario where there is none of them. Okay, before the eukaryotic evolution. For your clarity, I would say not only the eukaryotes form the multicellularity. There are even unicellular form. Even there is one. I've read the paper, but I don't remember the organism name. Bacteria which size of 9,000 micrometer size. It's like a big tube made of cells with a lot of nuclei inside. So, like your idea of chloroplast being inside aerobic, it could be a later event. Okay. But still, an anaerobic environment, because the experimental proof is just one example. Okay, it is not. That's what I'm trying to establish here. Not a single thing which is going to decide anything evolutionary outcome. It is going to be a multifactorial, and it is going to be a lot of things. Anaerobic is the evidence right now coming. As you see here, even the mixotrophs have evolved into a thing. Okay, and there is one more part which I have missed. If you pump like a crazy amount of oxygen, that evolve into even bigger thing. Okay, but still, like there is like lot of context is that we can discuss further. Jyotin? Hello?
Yeah, Shiva, can you hear me?
Jyotin, you can ask question.
Hello, Shiva, can you hear me? I think you have to turn on the speaker. Your side.
Okay. Okay, there is a question from Posh. Do yeast cells clump in any other stress other than oxygen? Like Ashwani have asked this question. Do ye like Ashwani for your question, like the clumping which is already predestined in this particular yeast which they have used for this experiment. So they have mutated this ACE2 protein, but which will mark the like the yeast are like attached to each other. So the aggregation or the clump formation is already fixed. So there is no separate like phenomenon which is here that like says that like any other stress because they haven't tested because already they are aggregate form prone. Jyotin?
Yeah, Shiva. Yeah, can you hear me?
Maybe Jyotin will be able to ask you later.
Okay.
So you spoke about oxygen driving evolution, probably, or lack of oxygen. And then after that we spoke about cytosine fixations. Maybe all these things seem to be considered as one multidimensional variable, so that we'll be able to understand.
Yes.
Do we have any answers to that? Like, do we know if there is any variable which can explain evolution better than taking just one, either oxygen or nitrogen or these?
I would say like you should like the evolutionary period which is happening. We are talking in the billion years of time scale. I would suggest like in our thinking process only, we should consider each and every possibility. Like when we are addressing any issue, I think possible. Aditya, that would be my straight answer. Like there is some other factor, and as far as I know, other than the oxygen and the size itself was a very important factor for the evolution because like when the oxygen is there, if you think about the oxygen which is present in the ocean, which is dissolved, if you take it to the consideration, that is way less than what is in the atmospheric oxygen. So there are so many factors, and how much deep the cells can sink. So many factors are there. Sorry, Jyotin, I'm not able to hear your question. Jyotin? Okay, you can type the question, or I'm not able.
Rahul? Datu bhai?
Is it already open?
No, it's not open. It's on the stock. Yeah.
Maybe he'll help.
It's yes. Start. ఏం కనబట్లా Good afternoon. Looks like everybody's had lunch, and I hope I don't bore you. So my presentation is on the topic putting 2 and 2 together. It originally started off as a pun, and now I realize that I have taken a very broad topic, so I'll try to explain as much as possible. To go to the pun, actually, so it's Tuesday, 22nd of February 2022, and I'm sorry, minimize it. So it's Tuesday. It's 22022022, which again is another pun because it's Tuesday, which is there are a lot of twos in today. But that's where it started, and then I thought I'll start talking about twos. So what is so great about 2 anyway? So it started off with it appears to be everywhere. We see bilateral symmetry in a lot of places in nature, and then we seem to have adopted it for a lot of times where this over here. So this is the view of. This is the top view of the Great Pyramid of Giza, and then there are various buildings. So it seems to be that we have adopted bilateral symmetry across cultures, across various places in design, in architecture, and every other place. And I started thinking, why is that so? But on top of that, though, we also have this kind of division between in cultures, ancient cultures and all, where we have like a heaven and hell, and then we have yin and yang, these kinds of concepts. And then recently I have also been looking at the way of binary thinking of people, that we think about it in terms of, let's say, this is feminine, this is masculine, and then we are recently coming into these talks about gender fluidity, and there's no binary, this kind of thing. The fascinating part of it was that we actually had to introduce the thinking of non-binary, the thinking of not thinking in the binary terms, which was the part that fascinated me. But I think we are still not there because it's not just about symmetry that I'm talking about. Even though today a lot of my friends have been making jokes about how I have to be, talking in terms of heat and stuff, but it's not just about symmetry. So let me tell you where I'm getting at over here. So if you take it, you take properties of any object whatsoever. All right. So we'll look at a rock, not this rock. So you take this rock, you take these properties. Let's say you take height, for example, or you can take anything, weight, strength, any other property which can be further divided into individual properties, and then you can actually make a a one-dimensional axis of it, right? So you take any property, there is you can make it one unidimensional graph, and then your object will have one one value which falls on that graph, right? So you take any arbitrary value which is either lower than the value of the object, equal to the value of the object, or greater than the value of the object. Right? So when there's only so you take a rock and then you strip it down to various properties, and then if you take every single property, and when you take only one property, then if the arbitrary value is the value of the product of the object, then the arbitrary object is the object itself. Unless you take multiple properties, if you take a single property, the object will, whatever arbitrary object is the same as your object. There's no difference. So as long as that is there, when you take only one property, if the arbitrary object is not your object, then there are this thing about arbitrary value being the value of the object goes away. So you are only left with either it's lesser or greater. So which also is the part which fascinated me, because it seems like when you take any single property, you either have it greater than or less than that any other property that you take. So we talk about it in terms of so something is greater than or less than or higher than or lower than, heavier than, lighter than, louder, softer, faster, whatever. So there are so when I'm talking about thinking about it in twos, I'm talking about not in binary, but in terms of looking at these things as in comparison also when we are talking about it in like higher values or lower values. Yeah, so this is how I do it. So, but why do I find this fascinating? So you take any point on any axis, it divides the axis into two. This is obviously a property. Now, if I tell this to anybody, they probably say that's the property, right? You have one line and you put one point, it will obviously divide it into two, which I understand, and I mean, like in all these years of existence, I understand that you have one line and then you put a point on top of it, and then it will always divide into two. But why does it actually have to divide it into two? Was another question. And let me tell you why this question is intriguing to me. Is it intriguing to anybody else at this point? So I'll try to explain what my conundrum is. So we'll take into consideration not all properties. We'll take one property, which is time. Okay, you have at any point of time, if you take it, every other value will be either before or after that one point of time, right? So it's just like any other property that you take of any other object. The reason I have taken time is because there are more theories which are explaining how time works. So you take any point, time equals t, time t equals 0. You either have the future, which is going forward ahead of time, or you have the past, which is going behind the past. All right. So the reason I'm saying that time is will probably be able to explain why I'm facing this conundrum is because we have something called block universe theory. We have various other theories. I'm talking about this one particular theory, where if you see on the top, I have one picture, and these four pictures are actually following a series of events. So you have T0, T1, T2, and T3, and basically the time is flowing from the top to the bottom. So whatever is happening in the first picture happens first, then after that the second picture happens, and the third picture happens, and the fourth picture happens, so on. But block universe theory states that, so I have just zoomed in the same figure. So all four are happening at the same time. So all four are happening at the same time. All these things are true and real at the same time, except that our brain is processing it in such a way that we make sense of it in terms of your one thing is happening earlier, one thing is happening later. Of course, this this has a lot of problems because this strictly adheres to a deterministic theory of the world, which some of my friends I know don't agree with. But basically, this theory states that the entirety of reality is like a movie reel. So when you look at a reel of a movie, so each of these is one frame. Okay, and if you play this frame after frame, it will look like this horse is galloping. But actually, this whole thing is part of one particular reel. Of course, the reel also is a two-dimensional way of looking at it. In fact, I think I should have mentioned this before. We do look at everything in a linear manner. Even this presentation, the PowerPoint presentation, which goes from top to bottom. It's not, it's only in one direction. So all of them happening at the same time, but our mind, interpreting it differently. I also found this cartoon. So like, what do we want? Acceptance of the block universe theory. When do we want it? There is no such thing as when do we want it. It's like there's no distinction between the past and present. Right? It's just an illusion, is what they say. Of course, so this theory is not without also, like I said before. So there are a lot of assumptions that we have to make. The thing I want, I'm not trying to propagate the theory. I'm trying to say that this theory actually gives us a new way of looking at how time progresses. Okay. In fact, time does not progress, and it progresses only in our heads. So can we look at every other dimension also the same way? Can we look at height, weight, smoothness, softness, anything, all these things, just like time, if all the time points can be put together as one, can we put all the other time points also, all the other properties also as one? And if so, does that mean that all the dimensions can exist at once? And if it does not, why does it not? There is one more thing one more theory that explains why our brains work the way they do. They are called skeptical hypotheses, that the world does not really exist and that we are only thinking about it. So basically, you guys are all hopefully listening. So this is your brain right now. So you are thinking that you are listening to Aditya's presentation. Maybe you're not listening. Maybe your brain is just in a vat and you are thinking that you are listening. It may be that you are, I'm standing on this podium, I'm touching this podium, and maybe it's not really happening. My brain is thinking that it's happening. So if the brain is actually thinking that it's happening, then in that case, could it also be that my brain is interpreting time in the linear manner? And also could it be that every other dimension that we are thinking about, every other property of every object whatsoever, is it all existent only in our brains? It's officially too much for me to elaborate on. So my questions are, like, if we are looking at things in twos, is this specific to our brains and specific to human brains, or it could actually be that it's only specific to my brain? Like, none of you actually exist. I'm not really sure. All right. So, but does the world look different if the brains work differently? I think I have made this presentation very short, of course. How will it look if the brains work differently? I really don't know. It's I cannot fathom how the world would be if my brain is not working like this. So instead, I thought I'll just go directly to the questions. But of course, I don't have any answers for your questions probably. So I'll just say answers, please. Thank you. Any answers? You should turn this on. Siva. Okay.
It's nice that you are trying to bring two things together. So, if you think in that direction, you are just thinking about two. Your entire point is actually not about two. It's about kind of any dimension you can think about. let's say instead of talking about the two, we talk about the VGR for the instance. So if you think in the same logic, like what the way I perceive the color might not be the same way you are perceiving. Like what I meant is like maybe the analogy you are trying to give is the two, but it could be any dimension for anyone's brain. Right? Like two is not necessarily the factor I'm saying. It can be answered.
So there are for everything that you take, whether you take an object or energy for whatever reason. So there are two kinds of properties to everything. There's empirical property, which exists regardless of whether you measure it or not, and there is something which is dependent. I don't know what it is called, but it's dependent on the observer. So when you talk about color, that's what it is. But when you boil it down to color, it actually is the wavelength of the color, which again is a sinusoidal wave, which is like you have the positive and you have the negative. So here also I can see twos here. So I'm not saying that, you know, like I am actually saying it. So I am saying that everything that we look at, it doesn't necessarily mean that there's binary bilateral symmetry. It doesn't need to be, but there does seem to be a fact that everything, when you put one point, let's say you say that you see what color do you see? If you see purple, and if I say that no, I see blue. So here obviously we are differing in terms of color, but what we are actually differing is if we boil it down to the the basic, then I am seeing a different wavelength than you are, which means that I am interpreting it as either a blue is what, like a longer wavelength or a shorter wavelength. It's a shorter wavelength than what you are thinking. So that's what I'm saying. So everything, it looks like we are looking at it in either a higher or a lower, as long as you boil it down to a single dimension, of course. If you do multiple dimensions, obviously there are more than two. It doesn't seem to be culture specific. It actually looks like everything. It actually looks like it's more like it's not even organism specific. I would say it's more like, I think. So, like I said, I don't really have any answers to you. It's more like so. I don't know till where I can go. Like, can I say that it is organism specific? Can I say that it's just the humans who are thinking about it? Or can I say that all the organisms on Earth that we know of are thinking about it in terms of like higher, lower, ups and downs and stuff? Or is it that we are perceiving them to be thinking this way? So my brain is in a vat as far as I'm concerned. Any other answers? Online, anyone? Okay, I guess this is done.
Thank you, Aditya. That was a very thought-provoking discussion. Next, we have Vedaballi speaking to us on when you change the way of looking, do the things you look at change?
Your presentation?
No. Founders. No.
This is the
No. No, not this one.
Shiva.
Yes. Can you just check if the mic is working for me?
Yes, it's working. I'll check from here.
Am I audible? I have to confirm this because of obvious reasons, you know. Okay, so I have a complaint that I'm not visible. Please. COVID precautions and all. So hello everyone. So the title of the talk looks a bit of a superposition of two different font styles, right? So if I were to make it clear to you, I have to choose one of this, and what you can see is that it becomes clearer. So the font style that you are seeing in the more transparent view, maybe it would be really spooky to imagine what if another version of me in a parallel universe chose that one instead, right? So is the conscious action of mind the key to deciphering the state, in this case, the state of the font style of the title of this talk? So by the end of my talk, I don't know whether you or whether I will be wiser about this, but I think it's worth a shot. So what is the fundamental nature of reality? How does space-time emerge? What is the true concept of consciousness? Science fiction has gone a long way to answer all these hard questions, especially the Infinity Stones theory given by MCU. But the real world is way a lot trickier than that. So till date, there is no single theory of everything that all physicists agree on. What all of them ask is the physical phenomena that we as conscious beings call reality is actually an emergent description from our perception of the true fundamental nature of reality. What does emergent description mean? Emergent phenomena are usually ones which have physical properties that are different from the properties of the components that they are made of from. The property of an emergent phenomena arises from a collaboration between the properties of the components that it is made up of. So like Aditya was saying, what if I'm touching this podium and it is not real? Maybe it's just an emergent description of, is the mic still working? Okay, yeah. Is it just an emergent description of what actually reality is? So here comes in a century of work on quantum theory. So William Harkins said that quantum theory is of fundamental significance in connection with almost all physical processes or all processes that we know. And recently, Sean Carroll, in his technical paper, which he has been supporting since long and gave an update in 2021, said that the true fundamental nature of reality is presumably represented by an abstract realm of possible natural events, which is described by complicated equations which define mathematical entities which are roaming through that quantum realm and actually are the components of the reality that we live in. So how does this quantum realm emerge to us as a classical realm? Because what we are observing as physical phenomena are all definite properties of a classical system where there is no state of superposition. So to get into that, first let us know what a wave function is. In quantum physics, a wave function is a mathematical description of the quantum state of an isolated quantum object, which describes all the possible distributions of its physical properties, such as velocity, momentum, and position, while the object remains in a superposition of states because of its particle wave duality. So the global wave function of a quantum system is given by the total wave function of all the objects that are included in the quantum system. So when we make an observation to measure the state of that quantum object, this wave function, which we can say as the property of the quantum object or system, seems to collapse according to one theory. As you can see, different states of the cat, and we observe only one, which is given by the Copenhagen interpretation, while some of the other physicists suggest that it does not collapse, but splits into multiple parallel alternate realities, which include us as the observer to be part, that is our wave function, to be part of those separate alternate realities. And this is a many worlds interpretation given by Hugh Everett and then expanded on by Stephen Hawking later through different propositions and theories. So is wave function now the building block that is the physical entity akin to reality? So objective collapse theory here comes in, which was given by Gherardi et al. In way old that 1986, which suggests that when an isolated quantum object is there, there is almost rare chance of it getting hit by any quantum event. But when the number of objects in that quantum system increases, there is a chance that there will be collapse of wave function of at least one of those objects, which will in entirety collapse the global wave function of the system. Why? This comes from the property of quantum entanglement, which separates the quantum world from the classical world. It proposes that two quantum objects can have their wave functions joined together into a larger single conjoined wave function, which will now simultaneously describe the physical properties of both the quantum objects, no matter how far they are. So it is not limited by the speed of light, and any action that is done on one of these objects will be conferred immediately as a way of information transfer to the other objects and define its physical properties as well. How does this form the basic of the classical world? So when we are making an observation as macroscopic systems for the state of a quantum object which is operating at a subatomic scale, we are always bringing in number of objects, whether we will it or not, because then the quantum object that we are observing is no longer in an isolated state. Here will be multiple other quantum objects that will come in collision with that object which we are trying to observe, and the quantum superposition state of that object will spread and ultimately get asynchronous and diluted as it spreads over the environment. In theory, this entanglement does not stop until it reaches the ends of the universe, if it is there. But what then is the key point that decides that this spread of entanglement will make our classical world into a deterministic one where only single choices are observed by us? This is given by something called quantum decoherence, which although the physicists have not been able to completely prove, but they have been able to go back to the states of recoherence, which is also a bit tricky. Quantum decoherence suggests that there is always a loss of information when the quantum entanglement spreads from this one isolated quantum objects over multiple objects, which again interact with multiple objects in the environment. In a manner, the quantum object and its environment get entangled. So when we are watching it or making an observation of it consciously, the wave function will always collapse. So there have been modern like improvisations to that theory, which suggest that it is not random hit, but the quantum objects actually interact with a random jiggling field, since in the quantum field theory, all particles, so to speak, are actually disturbances in some kind of field, like the shape of the electromagnetic field is given by the wave function of the photons. While Roger Penrose and Lajon Deleuze suggested that a quantum system collapses rapidly and randomly through the action of gravity. When a large number of objects are included in one quantum system, the mass inevitably increases. And since gravity is something that has not been quantized till date, so that force can alone explain why, which determines the collapse of this quantum system that fixes the position of this massive object in space and time. So how does space-time emerge? According to Einstein's special relativity theory, space and time are combined into one unified dynamic entity, which suggests that the future is deterministic, because space-time tells matter how to move and matter tells space-time how to curve, as aptly said by John Wheeler. But as Aditya was explaining the block universe theory, of Einstein, it proposes that in a block universe, which is separate from all other universes, it is also kind of an isolated system which does not interact with the environment. There are all possible, I don't know where the cursor is, sorry. There are all possible versions of an event that are available across different slices of that block universe, which different observers who are present at those specific space-time coordinates will be able to visualize, and it will be either past or future for them. So here in the case of an apple slicing, so an observer here will see the intact apple as a past event, while an observer along this slice will observe the slicing of the apple as a future event. This has also been explained in general relativity theories, which explains that there will be some phenomenon of time dilation between two observers who are traveling at different directions and different velocities across the space-time frame, such as there is time dilation observed for a spaceship returning back to the Earth, given like when you compare the atomic clocks of the Earth, and also on the mountain tops based compared to the sea levels. So what does all these actually imply? Is the future deterministic, as the block universe suggests? A new increasingly popular theory has come up, which is proposed by Lee Smolin, who suggests that the past is classical and the future is quantum. This means that the classical gives a definite, stack or shelf of information which you cannot change, while the future is still indeterminate, that is, you can say in a state of superposition, and hence it is akin to being quantum. Why? Because quantum and spread of quantum entanglement always includes the fact that there is an increase in information in the universe. That is, there is more collision between the quantum objects, there is actual complete spread of the quantum entanglement, there is dilution of the superposition states, and some of the objects are having their wave functions collapsed and are actually assuming form of a rigid physical nature, which we observe one state, not multiple states or superposition. And this increase in information in the universe is in coherence with the second law of thermodynamics, which suggests that the entropy in the universe always increases. Since the entropy cannot be decreased and the information that is already gained cannot be removed, hence time, the arrow of time, always flows forward. In this version of the universe, at least, it is not possible to recover the past states which have already have their wave functions collapsed or turned classical. So how does space-time then actually emerge? Since space is irrevocably or exchangeably referred with time. A recent experiment by Giacometti et al. This is a kind of a thought experiment, but they have suggested that if two observers across two different versions of reality, or say two different quantum reference frames, were trying to know their space-time coordinates, they would have to exchange an almost infinite number of objects, although here I have written n, which defines finite, infinite number of objects, quantum objects between them, and if they try to guess the spin state of that object, they might be able to determine the space-time coordinate of that object in the universe of the other observer. But while doing this, they will inevitably lose some energy. That is, gaining some information is always associated with some loss of energy. And since the space-time in the quantum field theory is always non-commutative, that means based as from the Heisenberg uncertainty principle, you cannot guess the position of an object fixedly if you know the momentum of it, and vice versa. So while you gain the information about one state of that object, you inevitably shape the space-time of that reality. So these two observers actually create a shared reality which they were actually trying to find out. So it is not a pre-existing background. They are making it up as they are trying to find what exists between them. So while the wave function collapse suggests an indeterminate universe, on the other hand, the wave function split, which gives the many worlds hypothesis, actually gives a determinate universe. It suggests that the wave function of all the multiple realities that are coming into existence are entangled together and are actually entangled sub-realities, not cleanly defined alternate realities, because the global wave function does not collapse. So they are all part of a huge wave function across all these realities, which keep on expanding as the realities multiply. That is, as many observations as you make. So what is this limit that defines the transition from future to past? Even if we know that an event has happened, as Aditya was saying, that how will we know if one event has already happened in the future and we are just observing one part of it? So different observers across different reference frames might be able to do that, but we won't be able to do that because it is limited by our conscious experience of that information gained. So is consciousness really the key to the mystery of reality? So what von Neumann and Eugene Wigner proposed when the wave function collapse theory was given, that wave function occurs at the instant of subjective awareness. That is the moment an observation is made by the experimenter. But till date, nobody has been able to explain how the wave function collapses, or why does the consciousness cause the collapse of a wave function. So many still do not believe that consciousness causes the collapse of a wave function. So what is actually consciousness? A new theory that has been given by John Joe McFadden has suggested that the physicality of consciousness is actually the global electromagnetic field of the brain. Why? Because previously what the physicists thought, that the consciousness must come as a metaphysical entity coming from the neuronal activity of the brain. That is, it is kind of an emergent description of the neuronal activity of the brain. Roger Penrose has even suggested that the consciousness is actually encoded in the superposition of microtubules, which are active during the synaptic firing in our brain. But what David Chalmers and other physicists agreed on is that the brain is too hot and too wet to be an isolated quantum system where such quantum superposition can exist. And if this quantum system does not exist, it would not be able to interfere with or get entangled with the observation that is required to collapse the quantum state of an object. So what the McFadden suggested is that the electrical firing of the brain generates an electromagnetic wave, which obviously comes from the particle wave duality of the quantum field theory, and information by the way of it being both a physical existence as well as an experience-based existence is also encoded in that electromagnetic field. This electromagnetic field has all the quantum entanglement ability to do all the quantum entangled properties with the environment, and in that manner it can affect the quantum properties of an object in the quantum scale that we are trying to measure. So just one thing before I leave the stage. Does non-local mind influence non-local matter? There has been a lot of spiritual and those kinds of thought experiments on it. But Dean Radin from the Institute of Noetic Science, who have been doing a lot of experiments on this through like almost a decade over now, have published preliminary studies which state that it's quite interesting actually. So what they told is a group of participants to look at a projector screen where photons which were entangled, that is coming from an object. You can make photons entangled from an entangled device if you conserve the momentum of while they were being created. So they were observed as a line graph on the projector screen, and they asked the participants to make the line graph go up while the moment it appeared on the screen, and just not focus on the line graph when it disappeared. So what they found strikingly, although it is preliminary studies, that there is a strong correlation between how the entanglement state of that line graph, that is the quantum entanglement photons, was affected with the degree of strength by which the participants put their focus on. So this is, although it needs to be proved on a great level, but it's an interesting study. So these are the words that I leave you with. Basically, I don't think that either you or I are wiser after this talk, but I think it was a bit of fun to delve into these matters, because ultimately what Niels Bohr said is correct. If you think you understand quantum mechanics, you really don't understand quantum mechanics. Thank you. Question? No. No, really, you're not.
I'm not audible.
Yeah, now you are.
So basically, what you are talking about the observation of a quantum phenomena. So there is well-known study that if you observe some quantum phenomena, that will perturb the phenomena itself.
That is what.
So how you, at last example, how you will observe some entangled stuff and try to realize this, because the way you want to observe the quantum state will be like that.
Actually, that is what the basis of it is. Because this is the concept of non-local mind. That is the whole thing that they're trying to prove. Does this non-local mind have the ability to change the state of that non-local matter, which is the entangled photons?
And another thing, when you're talking about quantum, it is the scale which is very small.
That is what
right?
I
It's
was.
in angstrom level, right?
Yeah.
But here, from the biological systems, I mean, millimeter or they're very big. So how do you correlate both the states?
You're talking this.
Because your quantum behavior will be not there, it will be decoherent, right?
So you are talking about in the perspective of the consciousness, right? If I'm.
Yeah, I mean, whatever you talk, it was macroscopic, right?
No, it was on how the quantum system, that was basically what I said, that how the decoherence brings about the classical phenomena from the quantum scale,
so
that is subatomic to macroscopic.
Everything will be decoherent, right?
Yeah, but the electron, if I am understanding it correctly, how the conscious observation makes an effect on the subatomic quantum object, if that is what you are saying, that is what McFadden has proposed, that since the electromagnetic field itself, like all kinds of fields, are explainable in quantum by the quantum field theory. So the entanglement properties, like the electromagnetic field of the brain, will be able to undergo entanglement with that quantum object that you are trying to observe. And this is what will be the basis of why the wave function of that quantum object collapses. That is how they are trying to explain it.
So,
yeah.
So when you are talking about the
what?
You
are talking about electrical field in the brain. How will it be entangled? No, I'm not talking about entangled. How will it be entangled by itself? The entanglement is, see, the electromagnetic field will act as a environment with which the quantum object gets entangled. That is what is the basis of the spread of superposition. That is what is the basis of the objective collapse, the spread of entanglement theory, which suggests that how the state of superposition gets lost while we transition from a subatomic universe to a macroscopic universe. But it doesn't mean that the two states cease to exist separately. It's actually a continuum. Thank you.
Thank you, Edbali, for your talk. I think my mind is really entangled now, right? So before it gets further entangled, I'd like to call upon the next speaker, Annapurna. The stage is yours. Thanks.
Oh, what happened?
should share it.
Yes.
Okay, hello everyone. Thanks, Shrikant. You requested for the session to get less entangled, but I'm sorry, I'm not going to be making it any easier. So I'm going to talk about hypnotherapy. So people who know me also know that I talk about mental illness and psychotherapy a lot. I mean a lot. So these are topics that not everybody responds very well to. I've seen responses ranging from blatant disinterest to wanting to run away. So it is with a lot of apprehension that I chose to talk about an uncommon type of psychotherapy. And I am running the risk of my audience either falling asleep or wanting to run away, but one learns to take risks. I am taking up a PhD after all, no bigger risk than that, is there? So anyway, coming back to the topic. So I'll start a little conventionally, going to the etymology of the word hypnosis. It comes from the Greek word hypnos, meaning sleep. And obviously, hypnos is also the Greek god of sleep. This fellow right here, do not ask me why he has a wing for a year, I do not know. So when I say the word hypnosis, what comes to mind? Probably a sweeping pocket watch, publicity gimmick on stage. Sounds very entertaining, but I hate to break it to you, that's not how it works. Hypnosis truly is actually a very tame procedure, and this is a better representation of what it looks like. And it's actually a psychotherapeutic treatment option. So how it actually works is a trained therapist would guide the client into a deep state of relaxation. And this deep state of relaxation is also described as a trance-like state. And even this is not all that uncommon. Have you ever zoned out while listening to someone? Hopefully that isn't happening in the audience right now, but we've all zoned out, right? So the hypnotic state is a little similar to that. Not the same, but similar. I'll come to that later on. So once a person gets into this trance-like state, they become a lot more open to suggestions and changes, right? And this is where the therapist can influence and help the person make changes. So I'll come to that in the next slide, right? So what hypnosis does is classically portrayed in a lot of pop culture. It cannot control an individual or make them forget the experience. They will certainly remember the experience. Neither can they be controlled by it, but they can be influenced and they can be, made to make changes in their lives. So what can hypnosis help with? What can it help treat? Apparently a host of things. It can help with relieving chronic physical pain, can help with insomnia and treat sleep troubles, addiction, weight loss, trauma resolution. And there is a lot of clinical evidence to support the utility of hypnosis for all of these things, right? So an example of how this would work is, for example, for weight loss. So usually people are put on diet regimens, but most of us are not very good at following them. So apparently what happens is in a hypnotic state, the suggestions that you're given to follow the diet regimen, to follow an exercise regime, you are more likely to actually take it to heart. Ironically, it's the brain that's involved, but as they say, you're more likely to follow it if you receive those instructions in a hypnotic state. Similarly, for chronic physical pain or for trauma resolution, a lot of times these are problematic because there is a strong association for these things in your brain, especially trauma, right? It's very difficult to forget trauma or to dissociate from it. So in the hypnotic state, if spoken or if given the right suggestions, it becomes easier to dissociate from the trauma and move past it, right? So a lot of clinical evidence for all of these things. More recently, people are also considering hypnotherapy for anxiety and depression. There is very less clinical evidence for this, but it's being worked on. So hypnosis can affect or help with all these things, right? So that likely means that it affects the brain. So how does it do that? I'll get into it. So for that, there are three major brain networks that are involved, right? The first is something known as the default mode network. So this network gets activated when a person is zoning out, right? When a person is not paying attention to the outside world, right? So I'll call this the zoning out network. Then the corollary, the central executive network, which is activated when a person is paying a lot of attention or doing challenging cognitive tasks. So this is the attention network, right? And then comes the salience network. right? So the salience network helps with deciding which stimuli to pay attention to. So at once your brain is receiving a lot of stimuli, right? Right now you're listening to me, there's probably a wind blowing, you can hear the leaves rustle, but somewhere your brain is filtering out. You are hopefully paying attention to me. So the salience network helps with that, right? So going into a little bit of detail of what brain regions are involved in these networks. So this is a little heavy, but bear with me, right? So this is the zoning out network, the attention network, and the filtering out network, right? If you notice, the regions that are involved in these networks are all cortical regions. So the cortex is something that's classically associated with higher order thinking, cognition, decision making, basically our personality, basically what makes us human, right? And what happens in the hypnotic state? So one major feature of the hypnotic state is a decrease in the activity in the anterior cingulate cortex. So what this means is that anterior cingulate cortex is something, since it's involved in the salience network, it helps in paying attention to the outside world and also deciding your mood according to it, right? So decrease in its activity would actually lead to a loss of critical judgment, decreased personal agency, and increased suggestibility. If I have to put it very crassly, तुम थोड़े बेशरम हो जाते हो if this particular brain region is less active, right? Another feature of the hypnotic state is an increase in connectivity between the dorsolateral prefrontal cortex and insular cortex. Both these regions are involved in body control, emotion, pain processing, self-reflection, and regulation. Again, this has implications for what the hypnotic state can affect and how it can be used as a therapy, right? And the third major feature is a decoupling of the zoning out network and the attention network. Now, this seems obvious, right? There should be a decoupling because in the hypnotic state, you're supposed to be zoning out, right? But in the normal zoning out situations, like what happens to most of us while daydreaming or something, your central executive network is still active. It is active to make you aware of your own self, of your own consciousness, if I may. So a decoupling of these two networks means that you become extremely focused in the hypnotic state and you actually dissociate from self. And this situation is actually defined as a different state of consciousness. I'll touch upon this a little bit further. So all these things happen during the hypnotic state and these give clues to how it may be useful, right? So most of these things and these networks that I've described come from the works of David Spiegel, who works in Stanford. He's a psychiatrist. Another person who's relevant to a discussion hypnosis is David Spiegel and most other modern day practitioners of hypnosis. Dolores Cannon was not a trained psychiatrist or psychologist. She was actually a regular Navy wife in the US who was born in the 1930s. But she established herself as a very well-known hypnotist and someone who practices past life regression. And this is where I go into entangling your brain, right? Bear with me. So how Dolores came across this concept was, as I mentioned, not trained in any brain sciences. She and her husband, Johnny Cannon, they started practicing hypnosis for simple treatments like to assist weight loss or to relieve stress, right? So sometime in the 1960s, when her husband was stationed in Texas, of all places, a local doctor approached them with help in treating a patient. So his patient, a certain lady, she had a serious problem. She had nervous eating disorder. She could not stop eating. She was extremely obese and had really high blood pressure, like really affecting her health. So the doctor was desperate to help her and he approached them that, can you try hypnosis on my client, on my patient? And the couple agreed. So Dolores and Johnny went out to perform hypnosis on this lady. And this was in 1968. So during the session, something really unexpected happened. So this lady was talking normally and then suddenly she took on a slightly different voice. And her personality, the way she was talking changed. And she started describing a life as a man in Chicago in the 1920s. That's when Dolores and Johnny realized that she's talking about the past, about a time when she was not born. That's when they realized, could she be talking about a past life, right? So it was, I mean, normally when you encounter certain things, such things, it's surprising, sure, but it's also low-key creepy, right? But they persevered, they kept trying it. And over the next several months, they worked with this lady, and she was actually able to remember, apparently, 5 of her past lives, right? And this series of events was documented by Dolores Cannon in this book called as 5 Lives Remembered, which was published in 2009, right? So after this experience, Dolores actually went on to work with several other people. And over the 1960s and 70s, she had more than 1,000 clients approach her from all over the world, right? And what was special about her work is that she did not just, help them resolve their issues by remembering past lives. She actually went back and correlated the facts they were talking about. If a person was describing a past life in, let's say, in some Asian country and were talking in a certain language, she actually went back to check if it fits, if the facts check out. So she has thousands of such case studies where the facts actually check out. So it doesn't appear that those people or even Dolores are blatantly lying about this. So what Dolores said is that, so we've had a lot of discussion about consciousness just now and what our conscious experience or what are, what we can fathom in our living life. So what Dolores said is that the information about our past life is probably not there in our conscious mind. So where is it coming from? The subconscious, right? So the subconscious is not a concept that was, introduced by Dolores. It has existed from the times of Sigmund Freud, right? It's called as a state of mind that's just under our conscious minds. It's there, but we cannot really access it. But apparently, during the hypnotic state, you can access it, and that's how you're able to remember even your past life, right? So based on this concept, Dolores developed a unique hypnosis technique called as a quantum healing hypnosis technique. It's practiced by very few people across the world, barely a few hundreds, and obviously it's very controversial. So I am not saying that I believe in this technique or even the concept of past life regression, but it's one of those things that you cannot easily believe, but you cannot easily dismiss either, right? But I will say that hypnosis itself could be a useful tool if done correctly, since I gave previous scientific hypothesis to explain it. So that's it. Thank you. I'll take questions if there are any. Okay, I suppose people's brains are too entangled to ask questions. Okay. Yeah. That is not working.
Hi.
Nice
talk.
Thanks.
So if it's not like they show in the movies, how do they induce hypnosis then?
So there's a lot of literature on that. Most therapists, I would shows her clients a series of images, and she basically is talking to them in a light manner and eventually pushing them towards the hypnosis. I should mention here that apparently hypnotizability is a feature, and different people score differently on that. There are some people who are highly hypnotizable and some who aren't. So that also is a factor. So from what I have heard from people myself and from reading is that there's only 20 to 30% of people who are highly hypnotizable. So there is a chance that you may even, decide to go for hypnotherapy, but you're just not hypnotizable. So it won't work for you.
How do you know if that person is hypnotized?
So there are certain cues that only a professional can tell that whether a person has officially entered the hypnosis stage. I honestly do not know what the physical or whatever the cues are. Thank you.
I'm not a firm believer of at least the past regression.
Yeah, me neither.
But I do believe that if you see many birds and even animals, for that matter, they have the instinctive behavior, like instinct I'm talking about. I'm not talking about the subconsciousness. Like if instinct is possible, which is like some things which is not learned but still we do. Okay, if that is possible, is that a similar mechanism could be, or even I don't know whether anyone has studied the instinct per se or not. It should be something to do with the brain, right?
I agree.
Something or past suppression or something kind of comes from the earlier generation.
So you mean something inherited memories? Yes. It's possible, right? But I don't know. See, these are things very difficult to explain. Like if I have to accept your concept that a person who's regressing to past life is actually remembering the memories of their ancestors. Possible. But how is it that they're like a woman who is alive in the 1960s, a middle-aged woman in the 1960s who didn't exist in the 1920s, is describing a life in the 1920s as a man? And that has nothing to do with her ancestry. So it's not something that's very easy to explain. And
that much is very possible. But I am talking about a very gross level of memory, because something we know that the instinct are being transmitted.
So there has been some research on how we inherit instinctive behavior. There's actually genetic to it. We can discuss that later. I have read a few papers about that.
Also again about many of us here have, but we have recently been talking about I don't know if it's you, but it doesn't necessarily have to be memory in terms of our brain. Let's say if there is some mark on my skin, or if there is even a mark on, let's say, if the road is wet and it's step on it after it dries up, that also is still a memory. And so, if there is a memory of that she was able to correct last time, it doesn't necessarily have to be stored in your brain. If that memory is, I really don't know where I'm going to this, but it's like there is some sort of memory somewhere which is being hidden, and it doesn't necessarily have to be something which you can bring. But then I started thinking about it in terms of how memory is stored in the brain itself. So do you think it's possible that whatever we are interacting with right now, whatever we are talking right now, is also stored somewhere as a memory and not in our heads?
Not in our heads. Where do you suggest there will be? I mean, it's an interesting concept, right? See, so a lot of people who diss the past life regression concept, they also say that it's possible that these are not actual memories. These are suggestions that the person's brain has interpreted a certain way and made a story out of it. Because, as they say, the brain becomes highly suggestible during hypnosis. So maybe they are memories, but they're not real memories, right? Still, all in our heads. So I cannot, for the life of me, imagine where else it can be stored. Maybe the concept that Veda was talking about, like a shared consciousness. Maybe.
So, what you're trying to say that we have subconscious mind that has been
What out, yeah.
But it can be a result of like the way memory is stored.
Exactly.
Think about the scientific experiments where people have shown that at least in the stage you can transfer the memories. And it may be the biochemistry of the neurons which has been lost the memory, and it's been now kind of similar.
Yeah.
And that's how now it is able to dictate that particular experience.
Yeah, that's actually what I was just telling to Aditya. It's possible. It's possible.
One argument that I have is you're trying to hit the part of it that is involving self-thought, self-witness. We live in a conscious self.
Right.
I'm facing a problem. I perceive consciously. Right? So I want to deal with it consciously. I want to put a self here. I want to drive myself into getting out of this situation, not in a subconscious. I want somebody to help me out, deal with that situation, that resonance with the reality that I'm facing.
Right.
Yeah, so I get your point, right? So I should make a distinction here. So past life regression is not a regular thing that comes up in hypnosis or its practice, right? So most certified hypnotherapists, ones who have degrees in psychiatry, would actually, address issues in your world or in your life. So of course, this does raise the question that you're asking, that I want to deal with things in the real world, in my conscious world. Why should I go into the subconscious or tap the subconscious? Hmm. Yeah.
I am a amplified sort of me, a desire in myself, and I can push on.
I totally agree, and that's what you're describing is basically cognitive behavior therapy, right? So certainly, I mean, a lot of people, despite having issues like even going through mental illness, are able to get past it by themselves. Sometimes you require the help of a professional to guide you to the process, right? So that basically is cognitive behavior therapy, which I would say 99% of psychotherapists follow. This is slightly different. This is for issues that people are not able to help with cognitive behavior therapy, something that involves deep trauma or extreme physical, chronic physical pain, stuff like that. But of course, there is not a very clear scientific understanding of it. What evidence that I showed, those just point at possible explanations. They are not actual explanations. So yes, your argument is very valid. It's an open-ended thing. Yeah. Thank you.
Thank you, Annapurna. That was a very interesting talk. Next, we have Pradeep speaking to us on functional outcome by mutational bias.
Okay. Yeah. So if people are not hypnotized and they are still in reality, so we can start. So the thing is, so there is a lot of biasness that is going on around, like some people are favoring something, some people are favoring the other one. So this biasness is not very truly bad. It is actually required. And this biasness actually helps you survive under certain conditions where when you are your true self, which cannot survive over there. So whatever this picture, you are able to see over here. So it just basically those biasness, whatever we have. So they actually allow us to evolve. And that biasness actually dictates whatever the descent that we were there. So it actually allows the incorporation of the modifications into that. And that modification, after modification, you will again become a descent, and that cycle continues. And that's how, where we have started from this point, which is a long period, and that we have reached to this particular diversity that we see. It is only possible because of the mutations and the kind of selection and like the way there are multiple things which have made us to be at this particular diversity. And if we see how this evolution is like going on, so what are the like key ingredients which are required for the evolution to work? So these are like three basic ingredients, like everybody is aware of, like genetic drift, recombination, and the mutations. And what we have perceived till now is basically that there is a selection pressure which actually gives a direction to these three, which are random. These three events are random, and this is the particular selection pressure which actually guides, okay, no, you have to go to this particular direction.
and that direction is basically leading to the increasing adaptation. It means that the selection is the one which is guiding the path. But if we try to understand this all things together, so this is basically, don't worry about this mountain. So this is a kind of fitness landscape where you just try to plot the fitness with respect to the genotype or the phenotype. So basically these are the functions. The fitness of an organism is being dictated by your genotype or phenotype. There's nothing new into that. So if you look around, like there are these three populations that we have, and you think that this green. So basically green is whatever the circles means this particular allele in the population, that particular allele is more frequently present. And whatever our conventional thinking is that this particular allele may be selected out. And if we try to plot it on this particular, maybe the black one was the one which was having very less fitness. And slowly this evolution is making it to move to the top where this particular green is being selected, and that's how it is being present more in the population. But there is something more to it that we tend to unsee it or tend to forget about it. So what I can say that if there is possibility that whatever the things that we are thinking are random, they are not at all random. So there is some certain bias that is associated with it. So what is going to happen? Suppose now we have the raw material. So for selection to occur, that is the mutations and all those things. Those are the raw material that we have on which my selection is going to occur. So now if there is a biasness in your raw material itself, so what is going to happen now? The selection that is going to pick, that will also be the biased because of the biasness in your raw material itself. So now if I say that the mutation which are happening over there, they are random, not random, they are kind of, some mutations are favored while some are not favored into the system. And that can be arise due to various, like you can play with the proofreading modules, whatever they are, like information, DNA repair or any translation like this information transfer pathway. And if you take like modulate either of the proofreading module, what is going to happen? It is going to increase certain kinds of errors that we generally think about. But why don't we think that it's kind of certain kinds of biasness which is required into the system? And that's how I can say that maybe this green allele is the one which is occurring more frequently than the other alleles. And that's how this is now being picked up my selection pressure. And that's how we are able to see it's more towards the tip of this iceberg that we are able to see over here. So now that like boils down the question that if that is true, that it means if whether the mutations are really random or not. If they are not, what kind of like the fitness or what kind of outcome that is being linked to it. So today I'm not going to oppose anything that this is true or this is not true, but I'm trying to give a flavor that there is a mutational bias that is there into the system, and that mutational bias actually impacting what is your functions over there. Just and this mutation bias is actually helping us evolve whatever we are today. So just simply take a simple example how like mutations can take place. So we know there are 4 nucleotides and this A, G, C, T, and there are only 2 possibilities that you can mutate. So there is transition. Transition basically means purine to purine and pyrimidine to pyrimidine. And there is another possibility is the transversion, where you can make 1 purine can be converted to the 2 pyrimidines. So what is going to happen? So if I plot if there is no biasness, what you're going to see over there, the probability of the transversion from purine to pyrimidines should be higher, 2 times higher. And that's how you have 66% probability that if there is no biasness, if it is random, you should have 66% that this transversion should be there into the population. So if we plot on a scale, so everything should be at this particular place. But when people have observed into like various genomes that they have studied, what they are able to see under various prokaryotic and like eukaryotic, that this seems to be biased towards the transitions. And this biasness is not same throughout. This biasness also have the certain biasness into it, where different species have different biasness into that. So basically the mutation bias is very common and it is being seen into the system, and it comes in many flavors over there. Fine. So now let's get to the real world example, like where this biasness might have helped. So this is one of the study that people have done, So it's like a simple phylogeny. It's not a complex phylogeny. So there was a group of people who try to study the group of birds. So the birds which are adapted to high, so this black one is the one where they are adapted to the higher altitude, and this is the sister clades of that particular. And this is not a single bird species. It's like the multiple bird species it is being seen. And what they are able to see that there is a kind of parallelism into the kind of mutations that is required to adapt to that particular higher altitude. So you can see that like suppose this was a glycine at that particular position, and generally when you are not adapted, if you are adapted, you can see that multiple species are having the serine at that particular position. So when we generally think about how this can happen, the canonical thinking, maybe the selection pressure is so strong at this place that this particular mutation is required at this place. But however, this group, they tried to analyze it a bit differently. What they did, so they tried to look, okay, from which particular like mutation at the DNA level it is leading to that. What is the genomic context of that particular? So what they are able to see that almost 50% of the mutation that is coming is basically there was a CPG island which is having the guanine methylated at that place. So what they are trying to say, maybe that particular locus is having a high probability of the mutation. That's why you are getting more and more raw material for that particular mutation to come into that. And that's how selection is more tend to select that particular mutation, which is now helping. So nature knows where to mutate you or where not to mutate you. It's kind of purging out the deleterious mutation. You know, oh, I should not touch this. I should be touching this part. So there is like another study that we all can relate. So it's like some doctors, they were trying to study, okay, they were giving drugs to the cancer patients, and what they were able to see that drug is not able to work there and the cancer is still growing. So they wanted to know what kind of mutations are actually going on there. So they sequence that particular biopsy sample, and so generally what we think that the allele which provides the maximum fitness, that allele frequency should be the highest. But when they did the mutation, what they are able to see that the allele which is providing the most benefit, which was present in the low frequency. However, when you see the mutations which are leading to the various alleles, they are able to see some alleles which are not at all essential for this drug resistance. They are very high in the mutation probability. So what they are trying to say that there are some positions in your genome which is mutating at a faster rate, and some part which is mutating at the very slower rate. So it's not only, we should not be worrying only about the selection. We should be we have to understand more about this mutational bias in order to combat this particular kind of situations that we are, like in this case, the cancer resistance and all those things. So these are like few examples I wanted to establish that there is a mutational bias is there. So people wanted to understand like why this mutation bias is coming into the system. So there was a study that they tried to analyze. So in 2012 there was a group. So they analyzed, there are around like 34 E. coli genomes which are different strains. The genome they analyzed, and they simply looked at the mutation rate. And similarly, what like earlier we have seen, there is a mutational bias. So if there is no mutation bias, we should see this mutation should be at this red shaped bell curve, and it should be between this one. But what they are able to see, that they are able to see two kinds of pattern. Some are very low mutation rates, some are very high. It means that there are some cold spots are there in your genome and some are in the hot spots. And when they analyze what all those cold spots and the hot spots, they are able to see that the genes which are highly expressed, these are the one which are the cold spots. Now it's very like mind-boggling question that has come over there is the gene where the transcription is going on very fast. And it means that you are opening up your DNA multiple times, and it is being already seen that if there is more like transcription, there is more likely chance that the mutation will be more in that area. But however, you are seeing a scenario where the genes which are highly expressed, they are having very low mutation rate. Now, how this can be possible? So when they looked into it, what they told that actually it's the transcription coupled DNA repair pathway that is activated over there. So it means that if you have more transcription going on, you have more of the DNA repair pathway will be coming. It means that when there is like DNA polymerase has done, so there is a basal rate, error rate that is there all throughout. So now suppose at one particular place, there are more of the error repairing enzymes are coming up. So they are going to repair the error which is done by your transcription, and also they are going to repair the error which is done by your polymerase also. So basically what is actually happening over there is that system is investing more in suppressing the mutations which are like not required at that particular place. Okay, but still that why, what is the mechanism behind it is still not understood. So recently there is one more study that has come. So what these guys have tried to understand, okay, what all the like genomic context, and they are actually leading to this kind of mutational bias. So what they did, they simply took a wild type plant and then they simply grown that plant for 25 generations, and they took in particular generation 40 seedlings. So then don't think that it is a very small number of plants. They took 107 of certain lines, so total of 40,000 plants they had sequenced, and then they looked for, okay, what kind of mutations are accumulating and what kind of things are associated with it. They simply wanted to understand it. So like whatever we have seen earlier, so they have just plotted with this, and what they are able to see that, okay, the mutations are there less in your gene bodies than they're your like normally non-gene bodies, like you have the introns and upstream and downstream region of the gene. They wanted to know how this is like happening. So what they try to do, they have done like they have accessed the GC content of that particular area. They have done a lot of epigenetic studies, and they wanted to see what kind of factors which are associated with the increased mutation or decreased mutation, and then they simply try to plot it. So what they are able to see that, so if there is more of GC content, that particular site will be the less mutation. It means that like that particular area, if we know that if there is more of GC content, it can be more of heterochromatinized area also. And also there are various epigenetic modifications that we know that they tend to decrease or they can increase your local density of the DNA repair enzymes also at that particular place. So this is how this, like there are various players which are there into the system, and they are actually dictating what kind of mutations will be there at that particular place. And it's not only the single nucleotide variations. They have looked into the insertion and deletions. That is also being guided by this particular, this is like well correlated into the system. So from this, it is like that, okay, there is a mutations which are less in your gene bodies, and there are high mutations which are there in your intragenic regions. And when you look, okay, now if there are like mutations which are there less in the gene body, it means that if there is, we think that there is no biasness. So there in that particular place, we should see at least in gene body, there should be uniform mutation rate. But what they could see that the mutation rates, there were less mutation when it was kind of an information transfer pathway, like the translation, cytoplasmic translation, splicing and all those things. Those are the genes where the mutation rate was very low. And the genes which are response to some kind of stress, like here mostly they're like hypoxia and various kind of stress. There the mutation rates are very high. And what they did, they took around 3,000 genes for which the essentiality and non-essentiality, all the phenotypes were not known. So they tried to plot with that what kind of mutations are associated with. They could see that the mutations are there less in the essential part and then the non-essential part. So there is a biasness. So these kind of experiments, whatever they have done, they have simply grown a plant with all the resources, whatever is required by the plant. There was no selection pressures at all. If there is no selection pressure, it means that the mutation should be random. If that earlier, whatever we have started with, but here what we are seeing that it is biased without any selection pressure. So this means that the mutagenesis is being tuned by your whatever the functional outcome is. So it's very simple understanding that if your genome is accessible to the mutagens means can be whatever the error system that you have. So if my gene, so if like whatever the biochemistry of the organism is there, which is providing a lot of reactive metabolites, or like there are various enzymes which does the mutations rate. So if your DNA is not accessible to that, how do you think that there will be more mutations? The one which is more in the periphery, that is the one which will be more affected by that. So there is kind of biasness. So there is, okay. So somehow it is being like well optimized that, okay, we have to have more mutation in that area. So we have to strategically place the various components in that particular area to have more mutation than this. So it's just a simple understanding where like you have like what is the genetic context. Like if you have more of DNA repair or protection pathways based on the epigenetic status, or like how well your DNA is bound to that particular place, it dictates the mutation. If there is more of it, obviously you will have less of the mutation rate that is being. So it So in a like if you try to put it together, so what you can say that it's kind of a, if you have a fair dice, so you have all chances will be coming like at a random, you know that the all probability 1/6 is same. But now if from here what I know that okay, mutations are biased in certain sense. What I'm doing, I'm reducing the probability from the random 1/6 to I'm not reducing it to, so I'm saying that I have already loaded the dice and I'm telling that this particular numbers are not going to come into that dice. So I have a more likely probability that whatever that number that I'm like expecting will be more. That's where the fitness is increasing, and that's how we are able to see all this mutation biases happening. So in a nutshell, so like all these kind of molecules which are there into, so they know their specific roles. Some kind of memory is being imprinted in those molecules that you have to do. So these so what kind of functions that they are going to dictate and help into the system. So that actually maintains what is like the delicate balance that we have into survival and the evolution. And if so, we just take an example. Okay, these mutations are random. If mutations are random, it means that essential part is going to mutate with the same rate with the non-essential. So in that, what is going to happen that there is a 50% chance that you are going to survive and 50% chance that you are going to die. So why I will have that kind of strategy where like I am having more probability of death. So basically I'm trying to minimize that this deleterious part and having more of it. So just the simple two take homes that the whatever we have understood till now that the mutations are random. They are whatever, like we are getting more and more evidence that yes, there is a variation at the sequence level which has some inherent bias. We are not at like completely random over there, and they are differentially susceptible to mutagenesis based on their genetic context, where they are present, what kind of molecules are interacting to it. So that's it. So any questions?
This biasness we're talking about each and every experiment. There was some kind of natural, not we're not adding any additional selection pressure. There are some conditions there.
Yeah.
So the biasness that we are seeing maybe simply because of the fact that this natural selection is. So basically, when there is a mutation in a very important gene, they may be at dead. So we are not getting that sequence in the when we are sequencing it. So that's why this biasness.
So what like you are trying to say that there is a purging of the individuals which are having the mutation in the essential genes. That's why we are not able to actually visualize them. So see, this question is really valid. But what I'm trying to say that there is a mutation. I'm not saying that those particular genes are not mutating at all, but the mutation rate in those particular areas is very low. The probability of mutation is very low because they are highly protected. Because in a system, we know that this particular territory, if I'm going to be, I am going to die. So the system has been optimized in such a way that I am going to protect those areas. But there is some like whatever the error rate that is DNA polymerase is going to do, that error rate will be there, the basal error rate will be there. But however, certain areas will be having more errors. Some areas will be having less error rates. That's what is a biasness.
And you are talking about the repair machinery.
No, not only the repair. So that's what I'm trying to say. See, repair machinery is the one that you can try to explain it. I'm telling that, okay, now in whatever the biochemistry that we have, so we have a lot of reactive metabolites are also there. Fine. So now it's the diffusive capability of that particular mutagen. So we know that whatever the DNA that is in the periphery, that is the one which is going to encounter it first. And you think about the DNA which is in the center. Do you think that that particular mutagen concentration, if it is being escaped from your other biochemical pathways, it will go and mutate the same rate over there? You're understanding what I'm trying to say? So in that way, there are like n number of possibilities with which you can have a mutation in your DNA. But somehow things are being optimized in a way that some reason I have to be being protected only. I am not going to touch them all. That's what, like in the see. Yes.
The variability bias trying to protect the protection and not like is also because of the molecular evolution that is going on, right? And the natural
Yes. See, I'm not saying that selection is not there. I am saying selection is there. Selection is the one ultimately that is actually leading to the path. What I'm trying to say, the ingredients that natural selection is getting, that is also a biased towards a better fitness. It doesn't want see, evolution is taking you to the next level. But if you are not able to survive in your own scenario, how you are going to go to the next level? So basically, this mutation bias is actually being optimized in such a way that I am not going to touch the survivability of the organism by not touching the essential components, but I will play with the components which are non-essential, more better. That's why if you have more variation, that you are going to survive better. That's why natural selection will select out those much better. That's what is the idea. Okay, thanks.
Nice talk, Pradeep. So, I also read this paper. So, this is a very nitty-gritty part of the experiment which they have done. So, just to clarify, for the previous question also, the mutation which they counted, they are checking the mutation or the sequencing being done before the evolution or selection can happen.
Yes.
Okay. Because there is no selection being involved. Already that bias you are talking about, I just wanted to clarify because the point was missed.
Okay, fine.
Because the selection is not even in the play here, because they are just collecting the seedling and doing the sequencing.
Yeah, yes. Okay, fine. Okay. Yeah, thank you.
Thank you, Pradeep, for your nice presentation. Next, I'm announcing Jyoti for his talk.
Presentation?
Yeah. Presentation. Just wanted to turn on the laser. Hello, am I audible? Oh. Okay, so probably this is the last talk, and the topic is tuning biochemistry through molecular crowding. So I have seen there is some seats or some places in this place which is more crowded, and some places are less crowded. So it already tells that there is some biasing when the molecular crowding is happening. Okay, so let's see about them. Okay, so basically, if we look at the cell, this is, sorry. Okay, so if we look at the cell, there are thousands of biochemical reactions are happening at simultaneously, and they has to be confined into some spaces, and also there has to be some kind of communication between them so that the cellular plasticity should be maintained or homeostasis should be maintained. But if you see at the level of reaction versus the level of organelles that are present in the cell, they are outnumbered. So that means cell has to develop some kind of reaction hubs where it can actually more localize some kind of reactions over there rather than
like even if it is in a confined space into some membrane organelle or not. So if we see the picture of that, so we can actually see there are many bodies or puncties that are present in the cells. Those are having some kind of specific biochemical functions, or I'll rather say there are some reactions that are confined in those places. So let's try to understand how these bodies are being formed in terms of energy and in terms of biasing, interactions. Okay, so if we have to make something crowded in some places, that means it has to be phased out from something else, something from the mixing, right? So now if we look at the entropy of mixing, suppose if you have two populations of like liquid or gas, they can freely move here and there, or maybe they have the Brownian motions or the thermal fluctuations. If you just give them to a divider and just put those gases or the liquids over there, they can be happily staying there. But when you remove this barrier, they'll try to mix to each other, and which will increase the entropy of the system, and which is more favoring. Okay, so good analogy is if we see our hostel rooms, it is more easy to mess with our rooms than make it more arranged. So you need more energy to arrange it. Okay, so now in terms of sorry. Okay, so if we see at the terms of energy, suppose interaction between two red molecule, which is a homotypic, is E of BB, and two black molecules, E of AA, and intermolecular interaction is E of AB, then average interaction energy of this homotypic molecule has to be greater than the heterotypic to make it demix from the mixed solution. And mixing will occurs in such a way, this demixing will occurs in such a way that it will slowly create a boundary from the mixed state to demixed state. And whatever is this, we called it as crowded of, so you can see the some molecules are crowded over here, homotypic, and rather than your mixed state. So this is nothing but the phase separation, and this is the transition that is occurred from this mixed state to this phase separated states. Okay, so now as I am saying that these are the more like interaction energy, what are the driving force in case of biology? So if you see the biological molecules, they are having non-covalent interactions, and there are prevalent non-covalent interactions that can actually cause multivalency to the systems. So now if we look at the rather simple systems where your poly G, poly T, or poly R rich region, we call it as low complexity region, they don't actually fold it into a proper tertiary structure. So one advantage of that is it will be very fluidic. That means it can adopt any kind of tertiary, any kind of structure based on its requirement. And if these interactions are occurring multiple number of times over there, that means it is increasing the valency. That means it is increasing the homotypic interactions. And this is the main driving force of the phase separation or liquid-like condensate formation. So now let's go for some examples where we can see them more. So let's see at in terms of biology what is happening. I'll take only two examples. So we all know the pure granules are very important for the polarization of anterior posterior. I think this is not I'm not able to. Sorry, I'm not able to play this. Okay. Okay. Yeah. Okay. So now you can see that these P granules are also the protein that present in P granule is labeled with GFP. Now you can see when the polarity in this zygote, when the polarity has to be determined, those P granules are dissolved from one region and go and condensed in another part of the region. Okay, so this is called dissolution. That means one part it has to be dissolved and another part there will be a condensation. So it is like you boil water in one place and then the vapor will go and condense in somewhere else. So, but this has to be in a very fine-tuned way so that this polarity should be maintained. Although the reasons behind this active dissolution and transport to another part to make active condensation is still not known. So it is very intriguing. So another thing is global CO2 fixation. So at morning, there is a talk from Shiva that oxygen actually plays a lot of role for multicellularity, right? So this same oxygen is actually playing role to phase separate some kind of proteins which we called as Rubisco. So if you know, the anaerobic Earth has more carbon dioxide than right now, but when the carbon dioxide's concentration going to decrease with more photosynthesis, with the time of more photosynthesis, then for if you take the enzyme as in a segregated way, it will not get enough amount of substrate to saturate. So there is a intrinsically disordered protein called essential for pyrenoid complex or EPYC1, which is actually having multiple contact points to Rubisco. And if you add this EPYC1 to the Rubisco, if you actually take these two protein together and you add it, immediately it condensates. And inside the cell also it is in a condensation state. And this is make a huge amount of network and huge density. So the main law, Arrhenius law, is your enzyme and substrate has to be interact to get it to make the product. So if your substrate is less, increase the enzyme as much as you can. Okay, so this is the some kind of two examples that I have taken from the nature. Like biological examples. Now let's go to the very early idea of protocell. So there is a recent publication that claims that if you have a polylysine rich idea and if you give the ATP, this polylysine rich idea can phase separate, because you can see the positive negative charge interaction will occur and over there they will condensate into a conservate kind of stuff. Now they are utilizing this to acting as a active conservate which can actually modulate the reaction. So now if you add phosphoenol pyruvate and that pyruvate kinase over there, it will actually transfer the phosphate to the ADP, and if your mixture has to have the ADP, then it will try to, as long as the phosphoenol pyruvate is there, the ATP formation will be there, and these conservate will grow and grow, and sometimes they will also, bud outs from the bigger conservate. So which is acting like a very early model of conservate that I will come little later. Oh, sorry. The same thing can be utilized for a functional reaction hub, where you can actually condensate not only one protein, but you can actually condensate many enzymes over there along with the gluing factor. So here the gluing factor is your polylysines unstructured region and ATP, and these hubs can actually now act as a active functional entity. So now, these are the some kind of synthetic approach that is going on over there to understand this phenomena. So now, what happened in past research? So we all know the Urey's experiment, where Miller and Urey, when they have actually take the inorganic molecule and try to get, I mean, they keep in such a harsh condition, and they get actually the organic molecules. Then Sydney Walter Fox actually, and at the same contemporary of Haldane and Oparin, what they have done, they pushed this experiment much further, and they got much more bioactive polymers, and they are much simpler, like polyproline rich, polyalanine rich, polyglycine rich peptides. Those are acting as a biopolymer, and they also have the same property of phase separation. And not only phase separation, they also perform some kind of basic biochemical reactions over there. So you can see this coacervate is made of from protein. This is called proteinoid. This is a polyproline-rich proteinoid, and this is made in high salt water. Like they took the seawater and they put all the raw material, and they perform the harsh reaction over there for overnight or few days, and they got this kind of conservate stuffs. And this is the electron micrograph of some proteinoids, where you can see they can actually bud, or they can actually aggregate together to make a reaction kind of happen. Now, I am in a, now what I am going to say that, okay, so as we are looking to this kind of studies, we're coming with that now more often when our proteomic studies have been done, like if you read few papers from the recent proteomics, there they have identified many these kind of regions, which is known as low complexity regions, and those regions are very simple in nature. So as we see, the more simplicity it has, the more interaction power it also have carried, and more likely to be phase separate. So now, in early probiotic soup is also having this kind of stuffs. So is it, does those seeds are being taken to the cell, and now we are seeing as a whole proteome, or it is just my imagination that I am thinking in that way? It is probably maybe the more adaptation through the mutational space. So thank you. No, it's not.
Yeah, this one.
You come.
This it.
So when
Yes.
So let us very much specific. So if you compare it to a cellular scenario like in vivo,
Yes.
and there is outputs of together, and if you have a same view for all kinds of protein, how it is going to work?
Yeah. So
specific for each protein.
yeah, so it's a very good question. So thing is that these molecular glues, or these, you can say these ideal regions, although they are simple in nature, but there are changes. And inside the cell, there is it's a highly crowded area, and not only one protein can comes into there. There are multiple factors that governs it. And yes, when you are taking, like, as biologist or biochemist, we take the reductionist approach. We take one to one and then try to correlate with another. But inside the cell, it is not always one to one. So it is like more number of interaction with different partners also can lead to this thing. And yeah, as you said, we cannot directly correlate from here to the cellular scenario. So any further questions? Okay, so is there any online questions? I mean, online audience is willing to ask something. Okay, thank you.
Thank you, Jaydeep, and thank you all students for their interesting talks. And I'll now announce a high tea break and we'll reassemble at 5 o'clock. Thank you.
Thank you.
Thank you.
I'll start the session then. Good evening, everyone. Today, for our last scientific interaction, we have with us Professor K. Vijay Raghavan, a distinguished professor and an eminent scientist in the field of developmental genetics. Professor Vijay Raghavan is the current principal scientific advisor to Government of India. He has started his scientific journey at Tata Institute of Fundamental Research as a PhD student after finishing his B.Tech degree in chemical engineering from IIT Bombay. He then went on to California Institute of Technology and worked there as a research fellow. He later joined TIFR as a reader in the year 1988. One can take inspiration from his ability to balance both scientific and non-scientific role in research. He played a vital role in establishing National Center for Biological Sciences under the aegis of TIFR in the year 1992. Among the various numerous awards that he has received for his outstanding contribution to science, just to name a few, he has been awarded with the Shanti Swarup Bhatnagar Award for Science and Technology, the Infosys Prize in Life Science category, and also the prestigious Padma Shri Award by the Government of India in the year 2013. He has also been member of several prestigious societies like the Indian National Science Academy, the Indian Academy of Sciences, the National Academy of Sciences, and the American Philosophical Society, among others. Professor Vijay Raghavan has served as the former director of NCBS and later appointed as the secretary of Department of Biotechnology, DBT, India. In 2018, he was appointed as the principal scientific advisor to Government of India. Beyond his lab science, he is also very passionate in making science accessible to common people. He is very active on social media and a proponent of frugal science. His interests spread beyond scientific research, such as institution building, science policies, and improving international collaborations to open new research opportunities. It is an honor to have you as a guest speaker among us for the CCMB Founders Day 2020. We are eagerly waiting for you to share your insights on this occasion. On behalf of CCMB, we warmly welcome Professor Vijay Raghavan for his talk. Thank you.
Thank you very, very much. It's a great pleasure to be here today, albeit virtually. I would have much preferred to be there in person. Can you all hear me?
Yes, sir. Yes, sir. We
yes.
can. Very well. Very well.
Good. First of all, congratulations on your Foundation Day. The CCMB is one of our great institutions, an institution we are all very proud of, and it's got great scientists who've done wonderful work over the decades. So congratulations on that. And there's a lot to be spoken about such institutions, how they were founded, how they grew. Something which I hope we'll have a chance to sit down in a relaxed way and talk about it sometime. Right now, what I would try to address is about how the world of science is changing. And in that context, well, rather, how the world is changing. And in that context, how the world of science must change. And I'd like to focus a little bit on how utterly unprepared we are for the dramatic changes taking place. And it's not just you as students or researchers, but all over the world, science needs to prepare for a new direction and a new approach. Why is that so? Very briefly, let me recount how we came about to the situation in terms of our understanding through science and technology, and how the world has changed through science and technology. Now, sometime about 10,000 years ago, as the domestication of plants and animals started, and the development of cultures of various kinds took a great step forward by the formation of cities, we saw dramatic changes in human approaches to aspects such as food, food, agriculture, and so on. The domestication of plants and animals and the cultivation required an understanding of seasons, of weather, of climate, and in many ways, the correlation between these and astronomy became very, very important. Similarly, as different societies started exploring the world beyond their narrow domains, using rivers and seas, again, exploration got linked to astronomy in a variety of ways. So a lot of the development of science was linked to economic growth. But one important characteristic of this growth was that it did not substantially interfere with the environment. In other words, it did not understand, it attempted to understand the extraordinary diversity which people saw around them. It attempted to understand nature's engineering, as it were, but it was not yet an effort at engineering nature on scale, even though there were, dams built and aqueducts and so on and so forth over time. This changed dramatically, most noticeably in the late 19th and the early 20th century, and this changed with the advent of the Industrial Revolution. Almost concomitant with the understanding which came through thermodynamics was the invention of the engine. And this resulted in the Industrial Revolution, which was therefore very tightly linked with fundamental research and applications in a manner which we still do not appreciate as much. Our understanding of light, later on electricity, magnetism, came very rapidly to applications in a variety of ways. But most important was the engine, which allowed people to make machinery which could mine and to commute and take goods from one place to another. Now, the growth which took place as a consequence of the Industrial Revolution had an important characteristic. And the characteristic was a simple one. You had the hubris that you could tame nature, that you could extract material from nature, you could shape it into whatever goods you want, you could have workers be involved in that shaping, and you could find market for the goods. In other words, you could have a better and better quality of life in a thermodynamic system which allowed lower entropy within that system, getting more and more organized, richer, whereas the outside of that system had more and more devastation and increase in entropy. This resulted, as time went on, at the Second World War, around that time, with the clash between imperialism, fascism in Spain and in Germany and Italy, and communism and its rise in the Soviet Union. This was a clash of various kinds of ideologies, but they came fundamentally from the desire to own and exploit natural resources and markets. That kind of an approach to nature resulted in an extraordinary understanding of nature's engineering. And over the 20th century, you had an understanding of biology, the structure of the DNA, of how life came about on this planet, Darwin and Wallace's theory of evolution, genetics, the late 19th century, 20th century, understanding through Mendel's work and later on. So not only did we understand nature's engineering through physical laws, understanding astronomy, but also in biology, and steadily in every aspect of science and through every aspect of science and technology, we also became nature's engineers. So from understanding nature's engineering, we became, as a human race, nature's engineers. That has resulted now, through the multiple industrial revolutions, into a very simple situation where we have moved from being survivors on this planet, yet another creature trying to survive, to actually being those who wield the paintbrush on the canvas of this earth, as the philosopher Dan Dennett has said. So the future of the planet rests with us. Now, it's all very well to say this, and it is true, we have brought the planet to a calamitous precipice where climate change, loss of biodiversity, environmental havoc, extraordinarily large, big cities whose populations we have to raise from poverty in a sustainable manner. All these are extraordinary challenges. But there's a fundamental change which is needed in our outlook, and that is simple, and that's a simple part, the complex part follows. In that more of the same kind of ingenuity which allowed us to survive now will not be feasible. Why is that? When we ran out of copper, if we could invent, fiber optic connections, why can't we do the same kind of thing now when we're running out of energy, have solar power, wind, nuclear energy, and have sustainable tools and carry on? Why doesn't more of the same not work? No, we need some of the same, but more of the same will not work because of the nature of the shape of the curve coming over here. Having more of the same means that we cannot go back in any way in having a sustainable structure and having, equitable development. So the complexities of climate change are such, of sustainable development and of ecosystems are such, that we do not know exactly from a theoretical framework what is it that we need to do, let alone what are the sources, what are the resources we can get to do that. So this is a very major challenge in science, which is how does one have an integrated, interdisciplinary view of how we can go forward in this crisis. It's all very well to say climate change is important, environment is important, biodiversity is important. But if you go outside on the road from CCMB and ask the common person, what are their top 10 concerns about their life, climate change will be number 97. They are more concerned about education, as you are, jobs, as you are, housing, health, getting good accommodation, and so on. So climate change doesn't figure that. So to them and to you and to the world in general, in addition to mitigation, adaptation to the consequences are more fundamental. Tomorrow, if there's an earthquake or a flood or a pandemic, what do I do? How do I stay safe? Is a major question most people ask. So we need to balance these twin challenges of mitigation and adaptation. Now, we all take great pride about how the world of science responded to the pandemic. And of course, there's a lot to be proud about. But it's also good to introspect and ask, is this the structure which keeps us in good stead going forward? And the short answer is that we need to change a lot in India, in America, in Japan, in Europe, everywhere. The consequence of the way we approach science today as scientists, as those in industry, as those in our university systems for teaching, all of that is late 19th century, 20th century approaches to science and to industry, right? Basically, we have taken a balloon and there are different spots in that non-inflated balloon of an approach to industry and how a chemical industry will grow, or how other industries will grow, how agriculture will grow, or how the university system will grow, or how national laboratories will grow. And now we have increased the number of these dots on that balloon, but we've also inflated it so that it occupies a whole new world. So therefore, we are in a situation where there's an extraordinary inflation in the numbers of institutions, the kinds of people in different kinds of sectors, but the qualitative nature of that institution has not changed. So our institutional structures the world over are not built for purpose going ahead. Our structures and particularly the life sciences is a major victim of that, are focused on the desire of individual scientists to excel. That's politely put. I would say it's focused on the desire of individual scientists to chase the metrics of excellence, pretend that they match actual excellence, and preen themselves as a consequence of reaching those metrics of excellence. This chasing of the metrics has resulted in great havoc all over the world. Let me elaborate a little bit. Around the mid-20th century, up to the 50s or so, the learned academies of the world had to elect people who they thought were absolutely fantastic. The numbers of learned people were few, and they elected very good people. They left out few very good people, and they made mistakes and elected some not so very good people. But today, the situation has changed dramatically. You have at the wall of excellence a very large number of people, and these are people of extremely high quality. So learned academies all over the world, if they elect people who they think are good, they're just touching, literally a few percent of excellence, leaving out most of the excellence. Let us leave aside the situation where they make errors and take people who are not excellent. And therefore, it is completely incorrect to associate excellence with those who are members of these academies alone. There are more excellent people outside than inside, far more. And therefore, it becomes responsible for those who are chosen, who have these metrics of excellence, to be representatives of their whole community and make that very clear. And that doesn't happen at all. Whether it is in India or in America, whether it's in terms of where you publish, the titles of what you publish, the H index or the citation index or the fellowship of academies, the chasing of metrics, chasing of derivatives as opposed to chasing of the real function has wreaked havoc on all science, particularly in the life sciences. When you ask young students what they want to do in their PhD after they enter, it is not that here is a very exciting question which I would like to address. Very rarely so. It is, I would like to finish these experiments so that I get my paper published in Journal X. And this is fueled by investigators who tell them that this is their route to success, because they themselves see this as a route to success. So this is a disastrous situation, and this has to change from within. It cannot come top down saying, thou shalt no longer chase metrics. It should be from below saying, we shall no longer chase metrics, and we should have mechanisms to evaluate groups and people which are different. Which brings me now to the next point. We have a situation today where a typical life sciences PhD takes several years. If you put the upper limit as it was many years ago as 3 years, everyone finished in 4. If you made the limit at 4, everyone finished in 5. And now when you make the limit at 5, many people go beyond 5. If you ask many people what their next step is, they are so exhausted by this long PhD that they give you an answer. Usually, there are extraordinary exceptions, and CCMB is an exceptional place. Usually, they will tell you they want to do a post. They don't tell you, here's an exciting area I've worked on in my science. I've shaped my interest in such a manner that going ahead, I want to do something different in this area. Some people do that, but mostly it's a post. So you have a 5-year or 6-year PhD after your master's. Let's say, how old are most people when they finish their master's? Let's say 21, 22. So you're 27 when you finish your PhD, and then you do a postdoc, and that's another, crazy situation. You have to get your paper in such and such a journal out, and you feel really terrible if you don't have that. And then, you're 32, and then institutes have this very interesting rule saying that you should be less than some age before they can look at you. And how do they look at you? Typically, an institution says, oh, this person spent a postdoc for so many years. Have they got these kinds of papers in these kinds of places? Some places are enlightened. CCMB is one. But most places are not enlightened, and they count the numbers of papers in the specific kinds of locations. But industry, again, is another challenge. Industry should welcome people with experience in research and explore. But industry has moved from being an engine of exploration of new kinds of science and technologies into commercialization, into being structures which are beholden to their shareholders. They are not beholden to their stakeholders. So pharma industry, other industry, want to be successful in making something which will sell, and the shareholders keep them on the straight and narrow. The stakeholders, i.e. the public, patients, and so on, don't have a major play in the way industry works, and that's because of obvious commercial reasons. So industry, on one side, an academic job has got all the stresses I said, but it lets you do what you want, supposedly. And an industry job is difficult to get, but it straitjackets you in multiple other ways. And industry, again, in the 21st century, is not as innovative successfully as it was in the mid and late 20th century, though there are exciting ways forward now. So this is the situation we are trying to address as a planet, as a country, 21st century problems, but our institutional structures are in the mid 20th century. Of course, there's another big challenge in India that all the top research institutions, such as CCMB, get top quality students from our universities after their masters, but they give rather little back to the university. So you have a situation where, the master's program may not be up to speed. So you have students and you teach them, and your teaching is confined to the students you get. They become better, and the university system and the research system is disconnected. There would be no significant difference in what CCMB does if it molecular biology of the University of Hyderabad. But it is not. It's not the Whitehead Institute or the Broad Institute of the University of Hyderabad. It is CCMB, which has a big challenge communicating with IICT, let alone with the University of Hyderabad. So these structures need to change. And if they don't change, we are going to be importers of solutions which are bespoke for other environments, which have no relationship to what we're doing, let alone solving the world's problems. We would be still very good. We would be exporting quality students whose ambition, whose fire in the belly will allow them to go abroad or to some institutions in India and elsewhere, which allow them to excel. But as a country, as a group of institutions, we are not likely to excel. But what is the good news? One doesn't want to be despondent and excessively critical on a celebratory event like a Foundation Day. The good news, first of all, is the ability to learn new science, new biology, new physics is something which is extraordinarily accessible today to everyone. And if you or I or anyone wants to learn something, when you're in the master's or starting a PhD, you can be 10 times better than your, mentor, your PI. Today, the life sciences requires an extraordinary ability to use quantitative tools of analysis, ability to dissect and handle big data, and also the ability to, theorize in a manner because there are new opportunities which were not there before. And therefore, the gap, as it were, between phenotype and genotype, how cells function and how cells respond to the outside world is something which is extraordinarily well addressable by individuals collaborating interdisciplinary and in teams. That is not clearly happening enough. There are some beautiful examples of that happening, but that's a really terrific opportunity, whether it's in epidemiology, whether it's in ethology, whether it's in ecology, whether it's in cell biology, or how the membrane functions. This collaborative approach is something which can pay off very, very rapidly. There are a very large number of bright people all over the world, so that's competition. But there are very few places even which can do this well abroad who collaborate sufficiently. So this absence of collaboration is a global matter, even though the volume of the scientific effort is extraordinary outside and not to be sniffed at. There's a great opportunity for Indian science to collaborate and go forward. Now, how then does one go forward from here? My suggestion, which is not easy to implement, it's easy to say, so I would say that's really an answer and not a solution, is that we need to reformat a lot of how our laboratories are structured. The labs, such as the CCMB, should, in my view, and all our national labs, NCDS, CCMB, IGIB, every group size of each investigator of one, namely just the PI. The PI then needs to persuade graduate students or postdocs to work on a shared problem. And you as a graduate student or a postdoc should be free to move around and work with anyone. So that's critical. You are no longer in the service of a PI in their pet project for their successes, but you are excited by the problem as they should be in conveying the excitement to work together. This, of course, is, again, as I said, easy to say, but it requires a change in the metrics of evaluation within your institutions and other institutions. This is the model which the Bell Labs followed. This is pretty much the model which the MRC Laboratory of Molecular Biology followed. And this is pretty much the model with some variations which the HHMI's Genelia follow. This allows individuals to move around hungry for problems which are exciting and solve them. Many of them will do reasonable work which will not address big problems, but that's what's happening now anyway, so it won't disrupt that. So you will get good training, that's not a problem. You will do decent work, you will write it up. But in the earlier stages of your career, where you are most adventurous and where you have a fire in the belly, you are not limited by what your PI asks you to do. And that's something which has to change fundamentally. Another thing which has to change fundamentally is it's not clear to me of what value a postdoctoral fellowship has. A postdoctoral fellowship didn't exist, it still doesn't exist in many areas of ecology and environmental sciences. It barely exists in engineering, but it's versioned in the life sciences into a larger and longer period, which extraordinary demands are made for, chasing the metrics. And then, you have new faculty members joining in systems across the world at a point of exhaustion. And they are then competing with each other for diminishing resources. Why not have people take on jobs which combine teaching and research right after a modestly long PhD, not too long, not too short, and then, develop their research problems, go ahead, collaborate, as I stated, and, Go on a sabbatical for 3 months or 6 months, whenever you want. The 20th century model is that you had to go to one of the great places and otherwise you wouldn't learn. That's no longer the case. The great places can come to you. You can go, sometimes for a short visit, sometimes for a longer visit, and sometimes on video. So this model also has to change, the change for a change in a postdoctoral setting. The last point is about our relationship to society and to industry. That too is fraught with a situation where all of us have great views on how society should be structured through science and technology, what industry should do, but we are not participants in that solution. As researchers coming out, I feel that structures like the CSIR should change its logo and have the S and the I intertwined with each other so that there should be no distinguishing between that. Otherwise, you may as well be a Department of Biology of Hyderabad University. If you are a national laboratory, then of the CSIR, then the S and an I must be intertwined. Now, that doesn't mean that each of you must interact with industry, but the institution should be interacting with industry, and maybe not even the institution. It can be a group of institutions of CSIR or CSIR with outside. But there should be an emerging property of high-level important interaction with industry. And this is something, by the way, is not new to CSIR. The National Aeronautics Laboratory interacted beautifully with industry and continues to do so. The IICT has done that a lot. The NCL has done that a lot. All of that needs to change into a more, 21st century viewpoint. IGIB and CCMB have done that a lot during the pandemic. But this needs to be a sort of an ongoing culture. It is an error to make a distinction between basic and applied science and academic research and industry research. There are just two kinds of research, good research and bad research. It doesn't matter whether you do it in academia, whether it's basic or applied. Just because you're working on tuberculosis doesn't make you a good researcher. You can be a bad researcher in tuberculosis or a good researcher in tuberculosis, and you can be an extraordinarily good researcher studying the replicase or a bad researcher studying the replicase. So what is the quality of your work? What is your contribution? What are the kinds of problems it is? So this is another kind of change that needs to be. So I'd like to stop here because it would be very good to, if Vinay allows and others allow, to get some feedback. I have, tried to suggest different kinds of directions. And as I said, these are answers to complex problems, but they're not solutions. Answers are easy to give. If there's a problem about, a road being too narrow, the answer is widen the road. But how to widen the road is a complex solution, which we have to engage with everyone around. So I haven't given you solutions, but I've given you answers, which is easy. And I look forward to hearing questions from you. Thank you very much.
Thanks, sir.
Little louder. Pradeep, little louder.
Hello. Hello, sir. Yeah, it was a nice suggestions that you have given to institution and like people all around. So my question is regarding that what you told, that is the postdoc is really necessary to actually get into the academia or like an industry, whatever you're saying. So what I want to ask is like what kind of like our government, like what kind of suggestions are being given to government, or based what kind of guidelines are being created? Because what we see, the kind of recruitments which are happening in institutes around us, and also in our institute or anywhere, we see that the postdocs is kind of at most essential criteria. And also what we see, there is a biasness that we see that outside postdoc is the more which is being favored, because the kind of statistics if we see. So what are your comments, how things are being suggested to the government, or what kind of guidelines are being created for that? That's what.
Thank you. You know, there is nothing in the guidelines for scientists and their career path which says anything about your having to have a PhD. All of those are internally, institutionally decided metrics. You know, the Indian Space Research Organization, their scientists, most of them don't have PhDs, and almost none of them have postdocs. Similarly with the DRDO, similarly with the DAE. So it's up to you to go to your DG, to your director and say, address these recruitment rules. It's nothing to do with the government of India. The government of India can't fix a problem which you have created or you don't think is a problem.
Hello, sir. Am I audible?
Sir,
Hello, sir. Thank you for such a wonderful talk. My question is, what I got from your talk is that there are opportunities for the institutions to merge with industry and have a change in the structure, but it is just what they are, or we are trying to maintain the status quo, and that is the problem. Or is there a problem at the level of the policymakers who determine how the structure of an institution should be, and the problem is there? It is not just about our old mid-20th century mindset of maintaining the status quo. What would you say the problem actually is?
I think policy and policy changes have an important role, and quite often more than policy, or in addition to policy, rules and regulations need to be enabling. But that said, I don't think institutions grasp the need of change, and they can learn from other institutions which have grasped that. 20 or 30 years ago, it was regarded as really extremely in poor taste for an academic to collaborate with industry in the West. CSIR led at that time of having collaborations with industry. Now, those collaborations need to increase a lot more. It's really an institutional mindset. If you look at IIT Hyderabad or IIT Madras or IIT Bombay, by the way, when I was introduced, someone said I was from IIT Bombay. I'm from IIT Kanpur. So it really, IIT Kanpur people get very upset if they're told they're from Bombay. Not anymore, but in the old days, Kanpur was considered the place to come from. I'm just joking. And this is not, this goes contrary to what I said about metrics. But anyway, the point is, if you look at IIT Madras and you look at their tech park, it took about 10 long years for them to come up. But now the culture of interacting with industry and having professors being involved in industry as opposed to their students is something very deep in IIT Madras. So there's an example right within us, in our own system, which many of you would say is bureaucratic, where something extraordinary has happened. IIT Bombay has done that. IIT Delhi has done that extraordinarily more recently. So this is something which, all of our national labs also need to grasp. It's much more difficult for them than for institutions which have a large alumni pool and large undergraduate pool. That's exactly what I'm saying. CCMB open its doors, or IICT and others, so that it is just bubbling and bustling with undergraduates from Hyderabad University, in addition to these extraordinarily talented PhD students which you have. That is the fire which one needs to light, and that's feasible.
Thank you, sir. Thank you for your answer.
Hello, Vijay. That was a very good talk. And this is Manjula. And I think what I'm going to ask you is maybe slightly a personal question. So, from an institute builder and to having a functional lab, now you are doing a lot of higher responsibilities, and which people think that being in government is a hard thing to do, right? Is a difficult task to do. How do you manage all this? Did you have any special skill sets, or it is, what is it?
Thank you, Manjula. Good to see you. Mentally, it is a situation, again, related to the points I made, that we have certain kinds of responsibilities as scientists, whether as students, PIs, institutional heads, or elsewhere. And part of it comes from the extraordinary generosity of support which we get. My own lab was funded, as you would say in Hindi, to makhi maar, to kill flies. And if the country is funding you, a country like ours is funding you to do Drosophila research, then it's important that you also, in parallel, at every stage, constantly think of how you can give back. It's very easy to get caught in doing what you're doing as the most important thing. Of course, it's very, very important, but there are other things. Once you start doing that, and I would urge everyone to do that in their own way at an early stage, and each one does it differently, and each one does it at different times, then the complexity of dealing with the environment becomes something which you're used to. And the key aspect in that is to express your views freely, but listen to others and consider the possibility that they are more likely to be right than you are. And that's something which science, of course, teaches us. But engaging with the government, with society, with politicians, with bureaucrats is actually a very thrilling exercise to see the diversity of views and the rooted concerns which they have. So it's been fun, and I've no regrets at all. And in many ways, I've been far luckier than I should have been, but it's been fun.
Thank you, Vijay.
Hello Vijay, this is Sivakumar this side. So as you talked about the really clearly about the matrices and how the matrices are changing with respect to the institutes, if not those matrices, like right now the institutes are dealing with the publications, the postdoc where you are doing and all those things. If not them, what would you suggest? How you can even choose the candidates which are very vast in number and what is your suggestions to deal with the non-metrical system of recruitment in any national level laboratories?
So I'll tell you what the alternative is and also the dangers in that alternative. By the way, a nice t-shirt. If you could ask your director to give me a large size one, I'd be very grateful.
Director, please give it.
But in terms of metrics, what we need to do is to look at institutions such as those in theoretical physics or high energy physics, mathematics, even in engineering. Now, there the publication metrics are very different, and quite often the publications come well after you've got your job. Most of the discussion about your value is through preprints. And your peer group nationally and internationally has a very good idea of what you're doing through your preprints. As long as you don't count preprints and you read them, and physics and mathematics and engineering in conference proceedings has done that for decades. There's no problem. It's not a complex problem. But we have, altered that, not only in life sciences, but in physics and chemistry and so on. So we say physical review research, the person must be bright. Cell, the person has attained escape velocity, right? And so on. This is not the way one can function. If you are putting your own money, your private money into research, right? Would you take a good person who you know is suitable and can address an important problem and solve it, over someone who's, you're clearly not a good person, but has got 3 papers in cell, right? You would say, no, it's my personal money, and I can see that this woman or man really has a drive, understands the problem, has got 1.5 preprints, but has got great potential, and let me take that person. But we have set up a set of measures which conflate precision with accuracy, right? We precisely will say that an H index or some other index is 14.362, and that's better than 14.2, and therefore we take that person. That's precision, but that's not at all accurate. You wouldn't hire a person in your factory by those metrics. Why are you spending government money hiring people by those metrics? Now, the danger, of course, is this, and that relates to the last one. You wouldn't do that with your own money, but people are very generous with someone else's money. They hire people on metrics on one side with government money, and now they'll hire, poor quality people with government money without the metrics. So we need to change that, attitude of being responsible. Now, that's a cultural attitude and not easy to fix by a process fix.
Thank you.
So based on your talk, I remember once you said that the more talented student after completing their masters, they target to going on like top research institutions in India. And basically what in a student's perspective, they know the research institution in India are very well funded compared to the university and the research in infrastructure and all. So recently when I was looking at the budget to 2020, as you are the scientific advisor to the Prime Minister, so from last year, the funding reduced by 4% to the science and technology. Okay, so when we have a scene like that in our country, how can we expect the scenario in the universities will be better in future? I know, because that are the deciding factors for a student or for a faculty to target a position in any institution. So after completing a like postdoc in like abroad or whatever, they come back and then they see that the research institution are in a better condition in terms of infrastructure and all. So they always go further. So how can we improve this funding, like funding for the country in science and technology? Because anyway, we are going to compare with ourselves with the developed country, the Western countries, and we know how much funding they get in terms of science and technology. So if we are to compare with them in terms of science, we should have that kind of funding. So what kind of advice or what kind of initiative that as a student or as a PI or as an advisor to the Prime Minister like are going on? Like, that is one of the question. Or how can we improve this funding structure?
Thank you. By the way, when you ask a question, could you also tell me your name? That will be nice.
My name is Suraj.
Suraj, okay. So Suraj, thank you very much. There's a lot to unpack over here. And it'll be a long discussion, but let me cut to the core. 90% of our research funding goes to research institutions and a few universities where only 10% of our students go. 10% of our research funding goes to places where 90% of our students go. So that's one point to remember. Of the bulk of our research funding, much of it is corralled by the Department of Space, the Department of Atomic Energy, and the Defense Research and Development Organization. In the West, these equivalent departments have 80%, 75-80% of their funding going to the university system, right? So it is not that if you are funding defense or space or atomic energy, you have to do it inside. You can still do it outside. There has been a major change, for example, in the announcement this year, that 25% of the defense budget will be spent outside DRDO laboratories. So while you're saying that there is a cut in the budget, and I'll come to why those cuts are if I have time, you actually are going to see a dramatic increase in the amount of money available in the ecosystem. That has happened also with space last year with the in-space reform. So these major agencies are going to see their money spent outside in the ecosystem. I think CSIR also should spend a substantial part of its research intertwining the S and the I, being an aggregator of talent, an aggregator of demand, and a provider of solutions, and it should fund bulk of its research outside its laboratories. Today, the bulk of CSIR's own research funding goes in salary, bijli, money, right? It is not a system which can sustain with a small component of research funding, the bulk going into infrastructure. Indefinite increase in funding also is not going to happen. If the economy improves greatly, there will be ways by which funding can be increased for purposeful things. Even when that happens, CSIR has to, or DRDO or DAE have to demonstrate that. But the fundamental problem which you've raised is a very serious one. How does one invigorate our university system? Now, the ISERs and the new IITs and the AIMS are some examples where things are happening. But you will rightly say, but these are new places. What about our 75 central universities? What about our hundreds of state universities? What about our thousands of colleges? Any sensible knowledge economy power would have research in these sectors. Our research in these sectors are minuscule. So research money has to go there. And then critics will say, oh, all very well, but the university system, the state university is riddled with bureaucracy, regionalism, blah, blah, blah. How can we do that? But there are solutions to that. One example of a solution which can be implemented in a new way in our context is the Inter-University Center. For example, the Inter-University Center in Astronomy and Astrophysics in Pune University. It was a fantastic center run by the UGC, which many people criticize, and it's done awesomely. Why is it done awesome? It's done because it's got a concept which recognizes its independence, but most fundamentally, it was started by Jayant Narlikar, right? So if you want to invigorate your state universities, each and every one of you top scientists in CCMB, in other labs, NCBS, elsewhere, needs to at some stage in their career do a Jayant Narlikar, right? By going into, in addition to your research into science administration, or going to Marathwada University or Osmania University or somewhere, nagging the University Grants Commission to start an inter-university center, which is much more closely embedded with the state university. Then instead of one cosmologist, you'll have 100. Instead of one cell biologist or structural biologist, you'll have 50. And you'll be populating the pool elsewhere. Otherwise, you will do extraordinarily well. And, you will be the world's greatest scientist in Malaysia. That's no good. You need to build up a school of many, many people. So that's something we have to embark on. So money by itself is not an issue. Per capita funding for research, if you compare with the West, India is not the kind of economy, because of its population size, is going to be able to do that any time soon. But by synergy and by interaction, we can make the current money go far. And when more money comes, we can do really well. Now, the National Research Foundation, for example, has just been announced. It was announced in a budget 2 years ago, and hopefully it will happen soon. That brings an additional 50,000 crores into the research budget, 40,000 of which comes by aggregating from all the ministries into shared projects, and 10,000 additional money. So when that comes, it's a huge input. But input alone is not enough. These kinds of new approaches are needed if more money is does well. You need more investment in the CCMBs of the world, but they should be quote unquote field stations for the university system, for students and researchers from there to come and work, just as Lawrence Berkeley Labs is, or Jet Propulsion Lab is. But that kind of an interaction is what is needed, not just getting students and doing great work alone. That also needs to be there.
Thank you.
By now I've forgotten your name. Subhash.
Suraj.
Sorry?
Suraj.
Suraj, sorry. That's a problem with a long answer. Thank you.
Thank you.
Hi Vijay, Somdatta here. Hi. I would go back to the thoughts on matrices, and you said that things in life sciences look much different from mathematical or physical sciences. And do you think that's simply because the number of people pursuing life science is so much higher, given that it's a more competitive field, there's more funding in it, I mean, it's like a whole vicious circle. So, of course, there's a mindset thing. I mean, people can always go through preprints and they can decide their recruitments accordingly. But do you also think that some of this mindset is driven by the sheer number of applications that people get in life sciences?
Thank you, Somdatta. It is a matter of size, but it's not the kind of size I think that you're saying. It's not, I mean, high energy physics, cosmology, astronomy have also, huge size of enterprises. It is the huge sums of money which go into the life sciences system, driven primarily by the US National Institutes of Health, whose budget is something like $35 billion, right? And big agri and big pharma, each of whose research budget is more than that of the US National Science Foundation. This big money has resulted in laboratory-based enterprises, largely in US universities, but also elsewhere in the world, which is the alpha investigator-driven approach, that investigators claim to be able to solve any problem. They will amass a lab of 30, 40, 50, 60 postdocs, a few graduate students, many technicians, and these are all, grist to the mill of their ambition and their hubris in solving things. This has become a self-fulfilling enterprise all over the world. This has led to a publication industry which is feasting on this attitude, and that publication industry makes billions of dollars of profit from our vanity in saying that if a paper is here or there, it's really good. This is a creation of us. The solution also lies with us. No government, no Lord above, no God can solve this unless we realize that this is something which we need to change.
Thanks.
Hi, Vijay. This is Shivranjani. So I have a comment and a question. So my comment is, your talk was very reassuring because I have done my PhD from CCMB and I think the infrastructure that we have is world class. And at the point I felt that I don't need to go out for postdoc. If I get proper guidance and direction, I can do my science here. But now this is followed by my question, which is, what all opportunities do I have here? Because as a job applicant, when the recruiters look at me, my competitors would be those who come from outside and they have publications. And as you pointed out, that publication, that's a separate topic. So where do I stand? Would I have enough opportunities? And are we actually in the state of accepting that not doing a postdoc from outside is okay? Or we still have few years to get to that understanding and accept that we do have a world-class infrastructure and the training that we get here is good enough if we get proper direction to go ahead with our career. So, thanks.
The problem does not lie with you. It lies with the interviewers and the interview committees. And should you get a job, there is a danger that you will become like them. And that is something to guard against. Typically, interviewers and interview committees, and again, CCMB is a notable exception, they count metrics and have prejudices. And it's bad enough if individuals count metrics and have prejudices. When committees sit, they then regress to the mean. They don't regress to the adventurous. They don't move to the adventurous. They regress to the mean. So this is a challenge. So the question is, how do you or I or whoever deal with this? My point is that there are two things which we have to do. One is to be, completely idealistic, clear on what we want to do, pursue that. But at the same time, it's very important to be realistic and understand the world, right? So you must therefore study your institution you're going to, your interview committee very carefully and see how your idealism fits there and see what you need to say to get that job. Your duty is to get that job, not to tell them about how idealistic you are and how you didn't go abroad or went abroad or came back or didn't come back. So that is a very important. So do your homework well so that you can deal with committees.
Yeah.
Committees are the worst invention in the world. Probably PCR is worse, but committees are close, in terms of artifacts. And therefore, you really need to study them carefully and deal with them. Now, second point is, we must all learn from what happened soon after the Second World War or during the Second World War. If you look at top scientists like Max Delbruck or Simo Benzer, these people started off their careers in very small or less known places at that time, right? In other words, if you get a job, take it and do really well there, and then you can move elsewhere. Mobility is not an easy thing in India, but that's something. If you do really well today, the world has also changed. They're willing to see you as a potential collaborator or someone whom they want to hire if you do really well. That many of those prejudices are going away, that you must be a descendant of some particular top-notch PI or something. Your work speaks for itself a little more now than earlier. So in other words, you have to be a good assessor of reality and deal with it. That's not easy, but don't lose your idealism while chasing them.
Thank you. I just hope I have enough opportunities in the future. I think I have crossed the first barrier for the time being. Yeah, thanks.
Oh, no, there are lots of opportunities, and we must move out of this mindset that you must go away and then come back. You can go to the, cross the boundary wall and go to IICT. You can stay in CCMB. You can go to Osmania University, Hyderabad University. In Hyderabad, there are 39 central government labs, right? And, do due diligence and make sure that, 6 of them give you top jobs.
Thank you.
Are there any questions from the online audience? If they have, they can type it in the chat box. Thank you, Professor, for being with us here today. As a token of our gratitude, we'll be sending you this small souvenir by post.
Wow. Actually, rather than that, I mean, this is fine since you put in some work. Thank you very, very much. But tell that man Suraj to make sure, Suraj's predecessor, to make sure that he tells Vinay to get me that T-shirt.
We have already planned to send you a T-shirt.
Wonderful. Thank you very much.
Thank you.
Thank you. Bye. Can I log off?
Yes, you can.
Thank you. Bye-bye.